All right. Am I audible and my slides are visible to all of you? All right. Thank you. So today, this is lecture number 6 and in our 5-day course. So very good morning to all of you. Yesterday, we talked about multiomics. In fact, from day one, we are trying to learn this completely new paradigm of treating cancer. And somebody asked me yesterday about this paradigm being useful even for preventing cancer. So I answered that yes, it is possible that we can have precision and personalized approach for preventing cancer, but still this whole concept is catching up because concept alone would not be enough unless we have the right tools to implement it. And that's what I'm going to address today. So even if you think something is right, but if you can't do it, if you can't show it, it's not going to be taking off. So fortunately, I would say that multi-omics is getting a lot of traction from the development in the technologies that are helping us to generate the data, to analyze the data, and come up with this paradigm of multi-omics application in precision and personalized medicine, especially for heterogeneous disease, multifactorial diseases like cancer. So I gave you the examples in my first lecture that how the self-driving cars were progressing. This was a fancy concept, a science fiction novel concept, but as soon as the technology was mature enough for collecting the data using the sensors, using the cameras, and they were able to capture 360-degree view of the street ahead of the car, this became a possibility, along with the computational power to analyze this data in the real time. So both data collection and data analysis progressed tremendously, and that made these self-driving cars possible. And now it's a reality. They are running on the streets of America, and soon you might see it in Jabalpur, in Aligarh, or somewhere in India as well. So it's not something that we should not think about because it is very difficult to do, but we should be able to even improve the techniques. So let's go into specific techniques that have been employed in multi-omics approach. As you can guess, that there are different layers of multi-omics. So there would be layers of techniques as well. Some of the techniques are common, but some of the techniques are distinctly different. So just a recap that we have got several layers. Here we have put just 4 layers: genomics, epigenetics, transcriptomics, proteomics, which are the main, in fact, the central dogma of DNA, RNA, protein. That is something that we focus on. And nucleic acid genomics is taken care of by the techniques that are similar. They are employable for both DNA and RNA. So genomics and transcriptomics will be taken care of by one set of techniques. Then protein is different, so we'll have different techniques of proteomics. And metabolites are also different, so there will also be different techniques for metabolomics. But due to some similarity between the two, they are commonly used, like you will see in subsequent slides, that there are some common techniques utilized for doing proteomics and metabolomics. So with the proteomics, the techniques mostly mass spectrometry. And in case of genomics, we are not talking about next generation sequencing, but there was a time when microarrays were very popular. And I myself have done a lot of work on microarrays in the US. So I know it was a lot of data that we used to generate, and it made a lot of sense, but technology took over, and next generation sequencing is now the standard these days. As I said, transcriptomics would be also employing the same technologies, next generation sequencing and microarrays, because the nature of the two is the same. Both of them are nucleic acids, so they will be using the similar techniques. For epigenomics, we also have sequencing, but there's some conversion that goes before uh, we do the sequencing, which is called bisulfite sequencing to take care of the methyl groups and the chemical groups that are modifying the DNA. And there's another technique called ChIP-seq, which is chromatin immunoprecipitation assay. So first we immunoprecipitate the chromatin using the antibodies, and then once we are able to do that, we do the sequencing. But this is not going to be for the entire genome. It is going to be specific for certain parts of the genome. So this is known as ChIP-seq. For metabolomics, we have got LCMS. So as you can see, metabolomics and proteomics. Can you see my cursor? Is my cursor visible to you guys or no? Okay. So as you can see, metabolomics is employing LCMS, liquid chromatography, mass spectrometry, and proteomics is also employing mass spectrometry. And here it is gas chromatography, mass spectrometry for some volatile metabolites that cannot be detected in liquid state, but in the gaseous state. So GCMS is going to be important for metabolomics. Now, again, I tried to emphasize on these things because we are doing multiomics because 90% of the cancers, they cannot be explained by mutations alone. I think yesterday talk and the other talk that you must have heard that this mutation in that cancer, that mutation in this cancer, but 90% of the cancers cannot be explained because this is not the work of one mutation or one DNA, rather it's a work of many molecules together. And subsequently, 75% of the cancer drugs can be ineffective in 75% patients. So mostly you would hear that this drug did not work because it's mostly hit in trial. So again, we have to emphasize the importance of using these techniques in generating robust data that can be analyzed for precision and personalized medicine. So this is a broad overview of how do we do multiomics based on the current technology that we have. So I think yesterday we saw how we did the sample collection from the surgery. So when you take out the sample from the surgery from a patient or say, for example, blood, it's not a homogeneous mixture of cells. Rather, it's a heterogeneous population of cells as denoted here by different color shades that the cells are different. This is all heterogeneous. This is known as intra-tumor heterogeneity. If we are talking about the tumor, solid tumors, it's intra-tumor heterogeneity. So this is a starting material. Now, this can be processed as a whole, which we call as bulk cell analysis, and this is the mostly employed technique because we bring the tissue, we crush it, and we take out the DNA, RNA, protein, but this is not from a single type of cell, rather it is from multiple type of cells. And we call it bulk cell analysis, and the expected data is something that is a representation of all the heterogeneous cells present in that sample. Technologies evolved, people started questioning that and I also told you yesterday about laser capture microdissection, where you can pick up these individual cells by looking into the microscope. And I have done that myself. It's a very tedious work, spending hours and hours in the dark room, looking at the microscope and isolating single cells. So that technique is also becoming obsolete because of the problems in doing it. So you can pick up these cells manually and then use them for extracting the DNA, RNA, and protein, and then do the work. But now with the help of single cell analysis, this is possible to sort them out. So you can see the different cell types and characteristics, they can be sorted out in different populations, and then you can use those populations for carrying out your analysis. So this will give you a better picture of the DNA expressed in these dark colored circles versus light colored versus the lightest color. So you'll have a fair view. It's like a high resolution picture, right? So if you have got a high resolution picture, you are able to see the picture even to a level of the pixels. And then there's something in between which is called spatial analysis. You don't isolate these cells separately, but you are able to say that this particular DNA or RNA or protein is expressed in a cell which is located in a particular location. For example, here I can say that the bottom right corner of the cells have got a particular type of DNA, RNA, or protein modification. This is known as spatial analysis, which means we are able to put them in context that where is this kind of cells located. Spatial uh, details are important because we have to know where the cells are located, which are causing the problem, whether they are on the surface or they are in the interior of the tumor. If they are in the interior of the tumor, the problem will be different. And if they are in the surface, we can target them easily and there will be a different strategy for killing them. So yesterday we got through the sample collections that we collected samples from tissues or animals, and then we can make organoids or cell lines. So these are the starting point for the subsequent technologies. We have to have the material in the form of cells, whether it is extracted from the patient tissue or from the patient blood, or it is taken from the animals from which we are studying, or we are developing the organoids, or we are talking about cell lines. So these cells are the starting material. And my aim would be to introduce you to as many techniques that are relevant so that when you end this course, at least you're familiar with what is available in this area in the market. If you go somewhere for your interviews, so you're not ignorant about these techniques, rather you have heard about it. But it will be very difficult for me to explain every technique in detail because I can tell you that each and every technique can be a lecture in itself. So I will be skimming the surface, but telling you what is the latest and what is not working anymore, so you have a fair overview of using these techniques for your own research. So genomics techniques, here is a little bit of evolution of how it happened. I think you might be familiar with the discovery of DNA and the double helix structure. And then the Caterpillars invented this PCR machine. So these are all laying up the groundwork for what we see today. So there was another breakthrough of the next generation sequencing in 2009. So you can see that the whole genome, which was sequenced in 2001, the next generation did not exist, next generation sequencing. It was done through classical Sanger sequencing. I'll tell you a little bit about that in a bit. So it was a humongous task done by several countries together to have the first draft of the Human Genome Project. And recently, this 2012 initiation of whole genome sequencing has started in UK in 2012, so almost years. And India also recently got a project where they are mapping 1,000 genomes from the Indian population. And so this is also now in the public domain. As you can see here, while we are progressing here, we are increasing the amount of data that we generate. Right? So the amount of genomic data that is created due to these technologies is huge. But analysis will definitely become difficult as well. So Sanger sequencing was easy to analyze. I remember even looking at the films, we used to do it manually. ATGC, ATGC, CGTA. So we were able to do the sequencing by reading the autographs manually. But with the increase in the technology, this analysis is becoming more and more difficult. That is another challenge. So I'll touch base upon all these in my subsequent slides. Starting from Sanger sequencing, this is the most basic type of knowing the sequence of the nucleotides in a fragment of DNA. So the simple steps are we need to first denature it, we need to separate the two strands. Then by using the PCR, we make multiple copies of it. We attach a primer so that we can start the color-coded or the radioactively coded nucleotides. So these are the 4 types of nucleotides, which are either radioactively labeled, so we can see them when we develop the film, or it could be color-coded, that can be done through the automated sequencer. Then these are grown through the polymerase reaction, and then we again denature, and then we subject these new chains to the sequencing. And as you can see that each one of these chains is going to be one base different. When you run them on the gel, they are all coming at the difference of one base. And this is how we read the sequence. So the sequence would be read as G, then again G, then it's A, then it's C, then T, then again G. So I remember using these autoradiographs. These were actually labeled and reading them from the sequence before the automated DNA sequences were available to us. So but it's not very old. It's like 20 years ago. The technology has transformed very fast and from this manual to the current level. Then we had the microarrays. In microarrays, it was based on hybridization. So on a single chip, which was a very small, 2 centimeter by 2 centimeter, we were able to put millions of probes. Probes were just the DNA sequences like oligos. So if you have a sample from a healthy cell or a pathological cell, you can just put them together and they are coded. And this coding would be seen in the form of the color. So if there is no color, that means it's not present in the cells. But if it is red, it means that this gene is expressed in the disease cells. And if it is green colored, it would mean that it is present only in the healthy cells. And if it is yellow, it means it is present in both cells. This is how we can see the presence of a particular gene in a particular type of cells. And this was very quantitative as well. So by the intensity of the color, we could tell that what is the level of expression of this gene or what are the copy number of this DNA in a particular type of cell. So this used to be, but there was a lot of background noise here when we used to do the hybridization. So this was replaced by next generation sequencing. The next generation sequencing is actually the extension of the Sanger sequencing, but at a higher level. In Sanger sequencing, you are able to do only 200 to 250 base pairs, and that's also for a small portion of the gene. If you have to do a gene of 1 kilobases, you need to do at least 4 to 5 sequencing. And that is also possible only one time. In case of next generation sequencing, we say depth. So we are able to do multiple sequencing of the same fragment of the DNA. And by doing this, we are increasing the confidence as well as the statistical prowess of the sequencing data. Because if you see here, if this data is done once, it might be wrong. There could be some technical errors here. There could be errors because of the polymerase. But if the same thing is done 1,000 times or 100 times or 500 times, it makes more sense. So that's what next generation sequencing does. So it sequences the same portion of the DNA multiple times. And also it can sequence several thousands of these fragments by using the different chambers, which are small chambers or small bubbles in which these are going on in parallel. So this is also known as the next generation sequencing is also known as massively parallel sequencing, which means there are thousands of sequencing reactions going on in parallel in different drop-like structures. So here's an evolution of sequencing technologies. Sequencing technologies have also evolved a lot, and it is continuously evolving. So if somebody from the engineering background or somebody from the technical background, they want to focus on the technology, this itself is a big thing to do. So like Sanger sequencing, it started somewhere in the 70s, and then it was spiral sequencing. Then there was iron tolerant sequencing. Then Illumina came in the market. Still the market leader is Illumina. It is having the best setup for doing the sequencing. But these all were limited because they were not able to sequence more than 500 base pairs at a time. And you all know that the human genome is 3 billion bases. So, and when you are seeing things in small fragments, then you're not seeing the complete picture. And it was very difficult to put them together. It's like if you have a ball of wool and you cut it into small pieces, and then you are asked to put them back together, you can understand how difficult will it be. You'll also feel like if these fragments were bigger, the task would be at least a bit easier. So with that purpose, the long read sequencing started. So long read sequences was done by PacBio and Nanoport. These are the technologies, these are the companies who developed the technologies. And they are now able to go into kilobases or megabases. But there's still no technology which can do the entire genome in one go. It's impossible as of now, and not even near to that, because if you see one kilobase or one megabase, still we are far behind in doing the whole genome sequencing. Right. So another important thing is when the human genome came out in 2001, it was a project done by the entire US government, along with many other governments of the world. So it costed millions of dollars. It was written here, it's $100 million for the sequence to be done in 2001. There's an interesting story about a private company. Craig Venter jumped in and they did it even cheaper way, but that's a separate story. But as you can see, with the passage of time, the sequencing economics have gone down. So it's now less than $100,000, and it's even some companies in China, they are claiming to do $100 genome. What it means for multiomics, that yes, now patients are routinely getting sequenced for their disease, and it's easy for the clinicians to order these, and it's easier for the insurance companies to afford it. So that's why now it's coming into the mainstream. In fact, in Iceland, they have sequenced their entire population. So, like at the birth, you get your Aadhaar card or some kind of an identity card. They get their complete sequence. This is your sequence. If you ever need it, this is going to be available. And this is a kind of summary for the various next generation sequencing technologies. So this is pretty comprehensive. You start with the common thing is the library preparation, where you start with a sample genome. You cut down this entire genome to small pieces and then put your own codes there so that you can identify it later on. This is known as library preparation. And then each one of them starts to go under sequencing in a separate bubble drop-like chamber. And then it can be either long reads, which means it's more than 500 base pairs, and it can be up to megabases. And the short reads, which is from 50 to 500 bases. And then long reads, as I told you, it's PacBio and Nanoport. These are the only two companies which are providing this service and equipment. So they are the leaders in their field. But in short reads, there's a lot of competition as well. But still, the Illumina is the leader as of now. There are other companies like Solid and Ion Torrent 454 sequencing. They all differed in how they were doing the PCR and the subsequent sequencing. So they are doing emulsion PCR, which means they made the small droplets in which they did the PCR, and nanoball PCR and rich PCR. So these short reads are still there. It's very much used, and it's still is being utilized in clinic mostly. But Illumina platforms are leading the game. So we have got this different variants of this next generation sequencing as well. I'll tell you about it in detail in a minute. But mostly you categorize them as, this is the DNA level, genomics, whole genome sequencing, and whole exome sequencing. Whole genome means entire 3 billion bases. Exome means the portion of that genome which is expressed, which is roughly 25,000 genes. It's about 1% of the entire genome that is expressed. So here the scope is much lesser, but you get more information, and because even if you sequence the whole genome, you don't know what is it doing. It's like dark matter, which has got no known function. So this data come out in the form of point mutations. You can find out small insertions or deletions. You can find out copy number variation, whether there's a increase in the copy number. Usually we have got 2 copies, but it could be 3, it could be 1.5, it could be 1.8, it could be 3.2. Structural variation, there could be a loss of an arm, like in case of colorectal cancer. Loss of 18q chromosome is a very significant step. And then you can also use it for lineage identification. All of us know about the DNA fingerprinting. That was used for kind of knowing where is the DNA coming from, and it is useful in forensics, paternity tests, and other things. Then we have got the RNA, and this is known as RNA sequencing, RNA-seq. I'll be dealing about all these things in a bit of detail in a few minutes. So the data that comes out from transcriptomics, that is in the form of differential expression, which means you get to know which gene is expressed more. Expressed more means having more numbers of mRNA, more copies of mRNA, because that translates directly into more protein, and which could translate into more function. The differential expression is some of the genes that are upregulated or downregulated in a particular disease condition, we get to know through differential expression. Then there are gene fusions. Gene fusion is there is a classical example in lung adenocarcinoma, NTRK fusions, where the one gene fuses with the other gene due to the chromosomal rearrangements. And this causes cancer. It's also common in blood cancers, especially in pediatric cancers. Gene fusion is quite common. We get to know through the transcriptomics. Alternative splicing is another level of complexity, because within the RNA there are exons and introns. So as all of you would know, that exons and introns are patches on the RNA, in the pre-mRNA, I would say. And before getting into the translation machinery, the introns are chopped off and exons are fused together. Now, this can happen in different combinations. I haven't put any slides here, but this is a separate topic to talk about. But alternative splicing generates different types of RNA depending on the need, which means we can have different proteins depending on the need. And RNA editing is there as well. The addition of the poly A tail or addition of a cap at the 5' end, these are all known through the transcriptomics. Then we also have epigenomics, where the addition of methyl groups can be studied. I just told you about ChIP-seq or ATAC-seq. They're going to tell us about the methylation patterns, if there is any modifications in the histone proteins, or if there's any modification on the transcription factor binding, because the transcription factors are the master regulators. They are like the people who are orchestrating the entire gene expression. So it's important to know about the binding of these factors, which ultimately results in differential expression or the functional aspects of the RNA. This is something I've just put it here, but I'll not be telling you in detail about this. This is a microbiome. I think I touched upon it a little bit yesterday, the day before. So we can also use the techniques to sequence the microbes that are living inside our body by using the 16S ribosomal RNA sequencing, or the shotgun approach or targeted panel. And by this we can know what bacteria is living within us, what fungus and what microbiome as in general is living within us, or we can have the antimicrobial resistance gene profiling. So in case of cancer, there's a strong evidence that is coming up that microbiome is disturbed. So we can know by knowing the change in the bacteria or fungi living inside our body, that what is the status of the cancer, or how is it going to affect the progression of the cancer. But this is not the topic of the lecture or the course, so I'm not going to do it in detail. And all these information that needs to feed into the effects, like what is the effect of this mutation. So we do the EQTL analysis, which is the quantitative trait loci, that what is the effect of this mutation on a particular phenotype, or we can do network and pathway analysis, because as I told you in the beginning, these genes, they crosstalk, they know how to find alternate ways of survival. So we need to understand them from the network perspective, that let's see how these genes are connected so that we can make sense out of their differential expression. If one gene is upregulated, probably the second one is downregulated, and both of them are coordinating with each other. This has to be known through the network and pathway analysis. And integrative analysis is the same when we are combining multi-omics and providing a full picture of the cell or the tumor. All right, so we go into a little bit of detail about the whole genome sequencing. So is everybody following here? Let me see if there are any questions. Because I'm not able to access, okay, no, there's no questions here. So you can speak out if you want to ask questions, because I'm on a separate tab and I'm able to see that. All right, so whole genome sequencing, as we just talked about, this is about sequencing the entire 3 billion bases. Now, why would we do it? Because sometimes you want to know the whole picture, irrespective of whether there's a known function of it or not. Okay, Madhya is asking, sir, can we use microarray data set for differential gene expression? Yes. RNA-seq is definitely better. Microarray is obsolete. So forget about the data that is there. You can utilize it for analysis. You can play around with it, but it's not as reliable as RNA-seq. So if you have to design a new experiment, go for RNA-seq. Microarrays are obsolete. I have done it. I know it. So that's why I'm telling you by first-hand experience. All right. So whole genome sequencing is about 3 billion bases sequencing. And most of it is having unknown function, but still we want to know, suppose we want to see the effect of the rest of the DNA, how is it changing, or if we just want to do it for characterization purpose, that we want to have the sequence of the entire tumor or cell, then we go for whole genome sequencing. But it is very expensive and it is very difficult to put together the entire thing. Again, just think about the ball of wool that you have. And if you cut it down into pieces and you have to put them all together, it's a much, much difficult task than if you're given a fragment of that wool and say, okay, take this one, cut it into pieces and put them together. So whole genome is like the entire ball of wool, and a portion of it is like whole exome sequencing. So I'm sure you would, all of you would say that whole exome sequencing is easier than the whole genome sequencing, and it is cheaper as well. But you miss the whole information. So that's the whole exome sequencing. And then there's also some targeted sequencing, because you don't want sometimes to go and see the entire exome or entire genome. Rather, you know that, okay, these genes are there, which we need to study. That is why we go for targeted sequencing. So in that, this is also a very lucrative work in the industries, that they are trying to develop panels. And I think I have a few slides on that as well. Panel of genes which represent a particular disease. So they have got panels for different types of cancer. They have got panels for different stages of cancer. They've got panels for different types of samples. So that is all through the targeted sequencing. The targeted sequencing is more like the Sanger sequencing, and but it is done in a high throughput manner. There is a comparative illustration of all these three. Whole genome sequencing, when we say reads, that means each fragment is sequenced 30 times. Reads is greater than 30x, because you're doing all 3 billion bases, so you can't go very deep. You can't do a lot of multiple sequencing with the same fragment. So it covers all genes and non-coding DNA as well, which could be structural DNA, which could be DNA with unknown function. Accuracy is low because, again, you are dealing with a big stuff, and it will also have a turnaround time is high because you have to make a big library and you have to do the sequencing, and then it will also cost more because it is going to be more labor intensive, and it will be involving more sophisticated instrumentation as well. Thank you, Asif. Whereas in case of exome sequencing, as we see here, we are only looking at the portions of the entire genome which are known to function, which are known to have RNA. The exon 1, exon 2. So we are talking about the exon level. So these portions are sequenced, not the entire genome. These gray portions, they are not done. So now we are more focused, so we can increase the depth, which means we can sequence the same fragment from 50 to 100 times. And this is only 20 to 25 genes. So accuracy is better. When you've got a smaller task to do, you're better. Definitely turnaround time is there, but it's less than whole exome, whole genome sequencing. The cost that we are talking about, the whole genome and whole exome, now it's in the hundreds of dollars. So it's more cost effective. And this is the depth, again, 50 to 100x. That means we are able to sequence each fragment 50 to 100 times. And targeted gene panel is something that we know, okay, we need to know not all the 25,000 genes. We are doing only 10 to 500 genes. So we are more and more focused here, and we can do the depth, it could be up to 500x. So the accuracy is increasing, right? Because we are doing the same stuff multiple times here. So we are more confident about this sequence. The accuracy is pretty high. Rapid turnaround time is few days. Now people can do it in a week time, along with the analysis. Because after you generate all this data, there's a lot of time and work that is in putting these pieces together. This is the analysis part. This is the work of the computational biologist. And to tell you they are the most highly paid because they are taking the data and they're putting them together and giving you the final information. So these are the three variants of next generation sequencing. So this was all about DNA. That the next generation sequencing is utilized for doing the sequencing of the DNA. So the first layer of the multiomics, DNA is covered through these approaches, right? So the second layer is the RNA. As I said, we'll be applying the same technique. Any questions so far? No. Okay, so we will move on. So RNA, the technique is the same. We are again sequencing every nucleotide there, and we are doing massively parallel sequencing. We call it as RNA-seq in general terms. This is somebody is talking about RNA-seq. It's transcriptomics, the second layer of the multi-omics, and RNA sequencing, or in short, we call it as RNA-seq. Right, so this is the entire genome that is known, 3 billion bases. Now, from the genome, the RNA or the pre-mRNA is made. The one that I was telling you that these are the dark red colors that you see here. These are the exons, and the light red color shown here are the introns. So the arrangement of these, suppose this is A, this is B, and this is C. So the arrangement of this could be changed depending on the need. So it could be A, B, C, or it could be B, C, A, or any combination. And this is just a simple 3 exon structure. If you look into some of my papers, there were 32 exons, 50 exons. So there are more exons, and that if there are more exons, it will create more complexity and more combinations. So once this is splicing is done, the intron splicing, the mature RNA is formed, which goes for the translation and making the protein. So RNA-seq is done for the entire RNA sequence, which is the pre-mRNA forming the mature mRNA, and then mature mRNA is digested to create the RNA fragments, and then it is converted into cDNA that's shown here in the blue color, and then these are sequenced. The double standard cDNA fragments are actually sequenced. I haven't put that slide for QRT-PCR. QRT-PCR also employs the same thing, but it is a different chemistry altogether. Okay, I'll tell about it later on. So here again, you come up with the sequence, and this sequence will tell you what was the sequence of the RNA. Right? So with this, we know that the RNA abundance as well as RNA sequence. So mostly people have been interested in knowing the RNA abundance, that how much of the RNA is there. The differentiated gene expression has been extremely popular. If you look into the literature, people get excited. Oh, this gene is upregulated in this cancer. Let's knock it down and we'll cure the cancer. But again, the concept of multi-omics tell them that no, don't get so excited. It's a bit early because you're not seeing the full picture. Right, then we have these fragments. We have to align it. This is all in silico work, so alignment needs to be done. Putting together the fragments of the wool that I just told you, that you have to put together those fragments to create the entire sequence, and then you can say whether it was splice variant A or splice variant B, because this sequence will be dependent on how this splicing was done here. So when you know the sequence, you will know whether it is BCA or ACB or ABC, whatever it was. So this sequence information will tell you which variant are we talking about. Then there is also a variation in case of RNA that you can do the whole transcriptome sequencing, which will be similar to the whole exome sequencing, roughly, because the whole exome is converting into RNA, and that's what the whole transcriptome would be like. And this will be known as whole transcriptome sequencing. So we take the mRNA, convert them into cDNA, and then you fragment them, make aliquots, and digest with the enzymes that are being used here. These are the TSP5091, and these are different enzymes which have got different cutting sequences. NIA3, MSP1, DPN2, and each company has got their kits available. They have got their own set of enzymes for making these fragments. And then once you have got the fragments, you can pool the aliquots and start to label them with the beads. And once you label them with the beads and then you sequence them after PCR amplification, and you can get the data about those fragments of the RNA. And again, here, Illumina is leading the industry by using its platforms, and you get good data. Here also, there's a multiple read. So you have got the depth, or you call it 50x or 100x or 200x. That's relevant for RNA as well. Now, we also have targeted gene panels, which means you are focused on a certain set of genes. If you look into the literature or if you look into the market, they have got multiple panels available. Some have got 50 genes, 100 genes, 200 genes, 300 genes. They will do it for you, and they will tell you out of that which gene is active or which gene is present or not present. This helps in the clinical decision making. So, but targeted gene panels is only useful when you know for sure that what you are looking for. If you know that, okay, I have to look for the status of the mismatch repair gene, say MSH2 or MSH6, you want to know the status because there's a cancer. This is that Asim was talking yesterday, hereditary non-polyposis colorectal cancer. We know that in that cancer, MLH1 is mutated. MLH1 does not work. So now with targeted gene panels, we just want to know about MLH1. So now we don't need to do the entire transcriptome. We don't need to do the entire exome. We want to know about the MLH1 so as to know whether this patient would be developing colorectal cancer or not. So there we go for targeted gene panels, where we are sure about a particular gene responsible for a particular type of phenotype. So it's more focused analysis. to run a bit faster here. Instead of sequencing the entire genome panels, they focus on predefined set of genes, and they get you actionable insight like HER2 and they are also easily doable, so they are available in the common market. Examples here are HER2, EGFR, MET, and then there are cancer-specific panels, as I just told you. They're available. Here I found one gene panel. These are the genes. They were utilized 1, 2, 3, 4, 5, 6, 7. 7 genes were used to classify 9,716 human cancers, and they found 28 molecular subtypes. So this is the gene panel that they use for patient classification, that which patient belongs to which subtype, so that they can decide upon the therapy. But our target is personalized medicine, going for a single patient, not the. So if I were to say this, these will be 9,000. Yeah, here. So 9,716 human cancers, they were subtyped into 28 molecular subtypes. With the personalized medicine approach, there will be 9,716 molecular subtypes. This is what we are trying to achieve. And as I said earlier, we are confident that the development of technology will be able to do that. Okay, so single cell genomics is another variation where we are trying to understand every cell as an individual, and single cell genomics has also progressed with the help of microfluidics. We can isolate these cells, and then we can do the sequencing of the genome or transcriptome of a single cell. So this is not a single cell genomics. And there's another variation called spatial transcriptomics. This is also a very upcoming field where you know which type of cells are present in what location of the tumor. So like if you look at this particular tumor, there are healthy cells, there are mostly cancer, these are the ones that are on the way to become cancer, they are very close to be the cancer. So this tells you which, and for the surgeons, you can guess which portion will they take out first. They will take out this red portion first, if they know, right? So this is what is being utilized for the spatial transcriptomics. So this was a benign tissue, but when they did the spatial transcriptomics, they were able to find no. Because if you take the sample from here, you will not be getting the full picture. Or if you take the entire tissue, even then you will not be getting the full picture. But with the spatial transcriptomics, you know where is the malignant cells. So with that, we move on to the next layer, which is protein. I have to rush. A typical single experiment in protein is still not there. It's far, far behind than the nucleic acids, what we have achieved with next generation sequencing. Proteins are far behind because in a typical single experiment, we cannot do more than 8% of the human proteome from blood or 35% from cells and tissue. So we are still not 100%. In case of DNA and RNA, we can do 100%. And this employs the technologies like chromatography and electrophoresis. Electrophoresis, it's pretty early. It's like Sanger sequencing of the DNA, 1D and 2-dimensional polyacrylamide gel electrophoresis using STS page or native page, or it could be liquid chromatography using HPLC or other type of separation techniques. Then we have got the mass spectrometry, which is the current standard that we use for proteome analysis, the mass spectrometry. There's also shotgun proteomics, which is more like targeted sequencing. So shotgun proteomics is identifying a particular peptide and particular types of proteins and just sequencing them for the proteomic studies. There's also technologies called SILAC, ITRAC, which are labeling the peptides and then sequencing them. So they use the radioactive techniques for labeling the peptides, and then they're able to sequence these. And then all the technology is also allowing label-free quantification because they use the time of flight, which is not here. So it's known as like Maldi-Toff. So they see how high the fragment flies, and based on that, they're able to characterize a particular fragment of the protein. So it's called label-free quantification. And this is complemented with the XRDs, X-ray crystallography studies about the three-dimensional structure of the proteins. NMR also gives you idea about the entire protein structure and how is it changing with any kind of mutation or anything. EASE2 hybrid tells you about the protein-protein interactions, that how these two proteins are talking to each other, because at the functional level, again, this is important to know. As we said in the DNA and RNA, we need to know the pathways. Here also, we need to know which protein is talking to whom and how, so that we can disrupt that or we can increase that interaction for a better therapy. And on the same line of the DNA microarrays, protein microarrays have also been developed and they have been utilized. They're still in use, but they are low resolution, high throughput screening of protein interactions. And so there are protein microarrays, and then you also have multidimensional protein identification technology, which is 3D level of identification of proteins. You can consider it as a spatial transcriptomics or a spatial proteomics, where you know which protein is getting expressed in what location. And since we are able to isolate a single cell, we can also do single cell proteomics. So single cell is nothing but isolating the cells from the bulk and then utilizing the transcriptomics or genomics or proteomics techniques from the individual cells. Here are some of the platforms available. This is Quantum C. It is for the single molecule protein sequencing platform, but the most popular is this one, Thermo Fisher's Orbitrap. It is able to analyze one sample in just 8 minutes, and it can identify up to 8,000 protein groups in a single run. But there are more than 100,000 proteins that are available at a particular time, so this number is still very low. 15,000 proteins can be done if you allow more time, and this also works for single cells. There are many other variations available in the market, but this is the one that is very popular. Next, we go on to the next layer, which is metabolomics. So we have got DNA, RNA, protein, and now the metabolites. So as I told you earlier, the techniques are the same: mass spec, GC, LC-MS. We can also do the ionization techniques. So we modify, we ionize these metabolites and then see their separation or their flight. And then we also have the NMR spectroscopy for detecting very sensitive changes in the metabolome. In case of metabolomics, you will often hear the terms untargeted metabolomics, which is again like whole genome sequencing. You're not looking for a particular metabolite, rather you are trying to understand the entire metabolites. And this is also done through the LC-MS. But you can do targeted metabolomics. It's like the panels of the genes that we talked about. It focuses on quantifying specific known metabolites. If you know which metabolites you want to study, then you have the panels for that, and you can do it again through LC-MS. So mostly, if you have got a lot of money, people go for untargeted metabolomics if they don't know what to look for. But if you know what the metabolites you want to study, then you go for targeted metabolomics. In my experience, we have a lot of computational tools. So we zero down upon which metabolites to look for by using the computational techniques, by using genome scale metabolic models, which can be done with the help of the RNA-seq data. So we don't spend a lot of money in doing the untargeted metabolomics. But yes, some groups might be interested in doing that as well. There are other omics, which is this list is also not complete. There are many more omics. If you look into the literature, you will find omics next to everything. So lipidomics, about the lipids, glycomics or glycoproteomics, because proteins, they get post-translational modifications. So this could be about the sugars. Then you have got the microbiomics, about the microbiome. You can use the metagenomics as well as metatranscriptomics. Interactomics, how these are interacting, the biological molecules, like protein-protein interactions. If you do it in a high throughput level, you call it the interactomics. Radiomics, if you are doing a lot of imaging, then you call it the radiomics. Fluxomics is another step. You're measuring the flow of these metabolites in and out of the cell. And if you're doing it at the high throughput level in a global perspective, we call it fluxomics. And single cell omics, I have already talked about. When you're talking about a single cell, you call it a single cell omics. Now, the most important part, and I have a few minutes for that, I would request Professor Hamid Ashraf to give me 5 more minutes, who's the next speaker, because all this is going to create a data deluge. A lot of data will come out. Now, if you are not smart enough to handle this data, this will be of no use. We need to generate information out of this data. And for that, we need to be able to analyze single omics. We need to be able to integrate them, and then we need to make the clinical sense out of it, right? And then we can use it for either disease subtyping or patient stratification. And don't forget, we have to look for a single patient. So this has to be done at the level of single patient one day. We can also use it for biomarker discovery that I'll tell you in my subsequent lectures that we discovered some biomarkers, we discovered some targets using these approaches. And we also can understand the regulatory mechanisms of the function of these genes. And ultimately, this can lead to drug discovery and development as well. Once we know the target, definitely we can know the drug that is going to act against it. So there are different strategies. We can do the integration. Integration means the integration of the genomics, transcriptomics, proteomics, metabolomics, epigenomics, how to put them together, the jigsaw puzzle. So this can be done at an early integration stage. So when you generate the data, you can start integrating at that stage, or you can even filter the data. You can clean the data and then do it in the later stages. So that's why we have got early, late, intermediate, or it could be hierarchical, which means that first we'll do DNA, then we'll do RNA, then we'll do protein. So this is hierarchical integration, which is more like a funnel shape. You start with a lot of data, and then you start zeroing down upon a particular type of proteins. And there are several computational methods used, like regression analysis, lasso, elastic. Bayesian methods are quite popular. Neural networks are quite popular. And these days, even multimodal systems are available for integration. I think I gave you some examples that Microsoft is working on, Google is working on, because these guys they're already very smart with the data. So now they're coming into this multi-omics arena, and they're trying to utilize their tools to analyze these multi-omics features. But the challenges are there. The data is very heterogeneous, coming from different patients, different levels, different stages of the disease, different cells. So this heterogeneity needs to be taken care of. It's like you are being asked to analyze a population of people living in a particular area and asking for what is their pattern. It's very difficult to see people moving in and out, and people having different habits, people having different shape and sizes, skin color, and so there's so many heterogeneity. It's very difficult to tell the behavior of the people from that. But yes, now we can do that, at least for the multi-omics. High dimensionality, each omics is a dimension. So genomics is a dimension, proteomics is a dimension, transcriptomics is a dimension. So we need to make sense out of these dimensions and give a target that can be worked upon, and the drugs can be developed, and it can be used for therapeutic purposes. Batch effects, which often happens in labs, that you do an experiment today, you do it tomorrow, it's not the same. This is called batch effect. So it should be taken care of that even one person does it and the second person tries to do it, they cannot do it. So these are batch effects, which is also creating problem for the data filtering and data analysis. And most importantly, data interpretation. Clinicians have to be trained for this. We as molecular biologists, biotechnologists, we have to make it as easy as possible for people to understand. And computational demand, which means you need a lot of infrastructure, servers, and the computational tools, the high-speed internet and high-speed processing that is needed for that as well. And this is my final slide, and this is in my this is a publication that we did recently, multi-omics for the cellular plasticity. I'm not sure if it is having a good resolution, but it's the same concept in everything that I've just told you. It's in one slide, that we start from a normal cell, we try to isolate the cell, we do the data generation, data storage is there, then we analyze it based on the genome, epigenome, transcriptome, proteome, and metabolomic sequencing and analysis techniques, with the use of artificial intelligence and other tools, computational tools. We can go for biomarker discovery, identifying therapeutic targets, mechanisms, and drug resistance, and personalized patient profile. And this is what my dream is, to one day come up with something that can be given to every patient in a personalized manner so that we can save lives. And the ultimate objective is to save lives. If I'm able to do it for one cancer patient, I'll consider my career fulfilling. With that, I'll say thank you, and I'll take some questions if there are. Otherwise, I'll hand it over to Dr. Hamid Ashra. Okay. Yusra Jamal, I think you should check your, if you were there in the yesterday's lecture, we have already posted the drive link, so you can take it from your colleagues. Saif Zafar, if a researcher wants to reduce cost and data volume while looking for specific mutations in a known cancer gene, would they be more likely to use whole transcriptome sequence or obviously target gene panel? Because when you want to do more sequencing, you increase the cost, you increase the data volume, you increase the complexity. You definitely go for a targeted gene panel. But again, you're missing some of the pictures. For discovery aspects, yes, you should go for whole exome, whole transcriptome, whole genome. But for the actionable items, you will need to go for these targeted gene panels. But Mavati, what is the difference between exome sequencing and transcriptome sequencing? Exome sequencing is for DNA, transcriptome sequencing is for RNA. That's a simple, straight answer. Exome is for the DNA, and transcriptome is for the RNA. Right? But yes, exome is that portion of the DNA which is making RNA. So if you do the transcriptome sequencing and exome sequencing, you might find similar results. But still, exome is you're taking only those portions of the DNA which are expressing genes, whereas in case of the transcriptome, you are seeing the alternative splicing as well. The exons might be rearranged, so you would find different splice variants. But in case of exome, you will not find splice variants. All right. If that answers your question. In that case, when should we prefer exome sequencing? If you want to know about the gene expression, you go for transcriptome. If you want to know about the mutations or copy number changes in the genes that are being expressed, you should go for exome sequencing. I would like to say your work seems very insightful and promising. I would like to ask, what aspects are you trying to cover using AI in your work, and how will it shape future research approaches? Okay, Sandeepan, just wait for my subsequent lectures. You'll get to know more about what we have done and what we are going to do. Okay, so if you are not satisfied with my answers, you can send them here, and I'll try to type it for you. Okay, while the other lectures are going on. Or you can send an email to the address that is communicating with you, and we will try to answer you through the email as well. With that, thank you so much for your patient listening, and I guess I'm not too late. Swati, over to you.
Yeah. Am I audible and visible? Okay, can you give me this access to share my presentation? Yeah. So my slides are visible and it's moving. Okay. Yeah, yeah. Assalamu alaikum and a very good afternoon to all of you. And I'm really thankful for that nice and kind introduction. And I'm also thankful to the whole organizing committee, especially Dr. Azhar, for giving me this opportunity. And I would like to make a, you can say, confession that this is first time I'm speaking on this topic. We do, as a necrologist, we do talk about a lot about different kind of tumors, especially in those small and tiny glands, including pituitary, sometime in thyroid, sometime in adrenals, sometime in parathyroid. But as an endocrinologist, diabetes, obesity, and thyroid disorders, they remains our bread and butter. And we don't look for, you can say, this precancerous condition or their risk for developing cancer in metabolic disorders. So this was a great learning experience for me to, you can say, how these things can, you can say, affect development of cancer in the patients of diabetes, obesity, metabolic syndrome. So in next 40 to 45 minutes, I'm going to talk whatever things which I could gather and I thought it is important to discuss with the audience. And as you know that metabolic disorders and how to minimize it. Yeah, I think. So metabolic disorders and cancers and what are the shared pathways and the dual benefit therapies and precision medicine in oncology. So you can say metabolism is something which we all have been taught since our class, you can say seventh or eighth in biology. It is the chemical reactions which is happening in our body to maintain the homeostasis, to drive our all the things which we need. So different kind of metabolic disorders, they are definitely there, and one of them is obesity and diabetes. These are two most common disorders we talk about. But if you look at from a different perspective, then definitely it's not only obesity and diabetes, but cardiovascular disease, neurological disease, cancers, and aging. They can also have a very important component of metabolic disruptions in their pathophysiology. And if there is something is there in the pathophysiology, then definitely that gives us a target for the development of, you can say, tools for the screening, staging, as well as management. The concept of cancer as a metabolic disease, they have very strong rooting in the early 20th century, and definitely this predates the discovery of oncogenes and tumor suppressor genes. And traditionally, we look cancer as primarily a genetic disease, which is driven by the mutations in the different kind of proto-oncogenes, tumor suppressor genes, and many times you can say epigenetic changes, which leads to uncontrolled proliferation. But there is an alternatively increasing and influential view which considers cancer as a metabolic disorders, which is rooted in the abnormal cellular energy and metabolism. And this concept has deep historical origins and recently it has regained strong scientific attention and backing with the evidence in the recent decades. And the metabolic overview of cancer dates back into the early, you can say, 19th century. In early 90s, in the early 20th century, in around 19, in the third decade of 19th century, a German scientist named as Otto Warburg, we all have must listen his name. and who has given this the idea of Warburg effect, where he observed that the cancer cells preferentially produces energy through anaerobic mechanism, uh, with the conversion of glucose to lactate even in presence of adequate oxygen. This phenomena, which is known as Warburg effect, was puzzling because normal cells typically rely on the mitochondria for oxidative phosphorylation, which is much more efficient. Uh, Warburg, at his time in 1920s, he proposed that the mitochondrial dysfunction was the primary cause of cancer, and the genetic changes were secondary consequences of the disturbed cellular respiration. But for several decades, Warburg's hypothesis was eviscerated by the rise in the molecular biology, because there is, genetics has taken a central, you can say, place in the development of different kind of non-communicable disorders in preceding decades. And the discovery of oncogenes, tumor suppressor genes, DNA mutations, and in cancer cells that shifted the dominant paradigm towards cancer as a genetic disease. And landmark findings in the tumor, you can say mutations in P53, RAS gene, BRCA gene, they development of cancer. And as a result, you can say that metabolic alterations were often regarded merely as the byproducts rather than as a causative agent. They were taken as bystander of product of the mutations leading to different kind of metabolic disorders. However, advances in the biochemistry, metabolomics, and system biology have reviewed the interesting metabolic theory of cancer. And now it is evident that the metabolic reprogramming is a hallmark of cancer, recognized formally in the updated hallmarks of cancer by Anahan and Weinberg in, you can say, the last one, I think it was in 2011. And this metabolic disruption, or you can say, disrupted, disordered metabolism is not only responsible as an individual, you can say, factor, but that also influence multiple other factors, which has been described by Hanan and Weinberg in their hallmarks of, you can say, cancer cells. And they're not only there is a problem with the disrupted glucose metabolism, but also there is lipid and amino acid and nucleotide metabolism to support the continuous proliferation, resistance to apoptosis, angiogenesis, and immune evasion. That disordered metabolism, they also play a very important role in these hallmarks of, you can say, cancer. And now many important genetic mutations are associated with cancer, directly affect the metabolic pathways. For example, if you take IDH1 and IDH2, that leads to production of oncometabolite, that is 2-hydroxyglutarate, which alters the epigenetic regulation. Similarly, the activation of PI3 AKTM TOR pathway that drives glucose uptake and anabolic metabolism. So these findings, you can say, they are bridging the gap between the metabolic theory and the genetic drivers, suggesting that they are deeply interconnected. As you can see on this, just a minute, there is pointer. So you can see that they are, deeply interconnected. You can see the cause and effect is difficult to tell, but what we can say at this moment that they are deeply, you can say, interconnected, and in bidirectional way. Genetic drivers, they lead to disrupted metabolism, and disrupted metabolism leads to problem in the genetics, leading to oncogenesis, or the progression of this. And from a clinical perspective, viewing cancer as a metabolic disorder has practical implications. This is very important because the diagnostic tools such as FDG PET, which we use to look for especially the, you can say, spread of the cancer, the metastasis, they exploit the increased glucose uptake by the cancer cells. And multiple targeting therapies of that which is affecting metabolism, such as glycolysis inhibitor, mitochondrial modulators, and dietary interventions like ketogenic diets, they are actively being explored and examined to the conventional treatment. So, being an endocrinologist, I cannot refrain myself from talking about type 2 diabetes because that is one of the of our century. Not only in this century, but also the last century. In the last 2-3 decades of the last century, there was, you can say, exponential increase in the number of diabetic population in the developed countries. But the problem now is that the middle and lower income countries, they are seeing the most, you can say, they are most affected by the diabetes mellitus rise in the recent decades. So it is increasing day by day in our own country and in our own city. And definitely you can see this 589 millions, almost 600 million adults worldwide are having diabetes, and this is the most recent data published by the International Diabetic Federation just few weeks back. And cancer incidence is, you can say, it is also increasing very rapidly, and we all are discussing different kind of theories about that why it is increasing. And type 2 diabetes increases the cancer risk by, you can say, 20 to 40% across multiple cancers. And diabetes and cancer, they are interconnected through shared risk factor and the pathophysiological mechanism. As you know, type 2 diabetes is characterized by insulin resistance, hyperglycemia, increased risk of developing certain types of cancer. Notably, individuals with diabetes, they have very high risk of liver, pancreatic, endometrial, colorectal, and breast cancer. As you can see that the 2 to 8-fold higher risk of developing these type of cancer, and also breast cancer and bladder cancer. The risk of liver cancer is approximately doubled in patients with diabetes, partly due to non-alcoholic fatty liver disease, which we will see in coming slides. Pancreatic cancer is elevated due to problem in insulin production, stress, chronic inflammation. Endometrial cancer increases the risk because of hyperinsulinemia, which stimulates the cell proliferation. Because of insulin resistance in type 2 diabetes, in the initial years of diabetes, there is increase in the insulin production to overcome the insulin resistance which is there inside the body. So these, as you know, insulin is anabolic hormone. So that leads to proliferation of large number of cells across all over the body. So that definitely is going to increase some risk. The shared pathophysiological mechanism include chronic inflammation, oxidative stress. Oxidative stress is very, you can say, you can say one of the major pathophysiological mechanisms for development of different kind of complications of diabetes. And if you look at the epidemiology of diabetes, this is the most recent data which we have collected from Aligarh, in and around Aligarh. And this is the data. We do have data of more people, but they were more than 40 years. We have not included that in this analysis. But you can see the diabetes in young, it is increasing very rapidly, especially in our country and more so in our region. You can see that this is a population-based study where we have included people from the rural as well as urban area. You can see that in patients, in individuals who are less than 40 years of age, diabetes was present in 15.2% of the patients. And pre-diabetes was also very high, it is 27.6%. So you can see almost 40% of our population has dysglycemia. And male and female, they were, you can say, equally affected almost. The reason behind this is multifactorial, but the issue with the young onset type 2 diabetes is that, as I was discussing previously, what are the shared mechanism of development of cancer in patients with diabetes. So, the complications of diabetes, they are the factor of, multiplication factor of the degree and duration of hyperglycemia. So if a person is developing diabetes at the age of 60 years and he lives for next 10 to 15 years, so duration is quite short. But if a person is developing diabetes at the age of 30 and he has to live till the age of 75, for last 45 years, patient will be exposed to hyperglycemia and these problems. So development of complications will be very high. So this is hitting very early. Diabetes is hitting very early in our country nowadays, especially in our area. And if you calculate this ratio of diabetes to pre-diabetes, for example, from this data, this is more than 1. So if the data of diabetes to pre-diabetes is more than 1, that means that in next 5 to 10 years, we will be having a tsunami of young onset type 2 diabetes mellitus patient in our country, because these pre-diabetic individuals, they will convert into diabetes within few years. So we need to look into not only, the diabetes part, but the other parts also. For example, as I am discussing today about cancer, so we need to look for those things and how to we can, prevention is always better than cure. So we need to look at the data, we need to look at the risk factors, and at least we need to apply some kind of, you can say, measures to prevent the complications, including the cancer. This is our another data. Here you can see, we have included, this is also population-based data along with our community medicine colleagues. So you can see that around 511 individuals. And so whenever I complete, my time is up, you just let me know so I can rush through these things. Total 511 individuals with first degree relative with type 2 diabetes. So any person who has got a family history of diabetes in the first degree relative, including the uh, parents and first degree cousins. So here you can see the dysglycemia is almost 50%. So if you have any person in your family, immediate family, who has got diabetes, then risk of diabetes is very high. And if you look at the, you can say, less than 40 years of age group here also, so you can see almost a very good number of persons, they are also having pre-diabetes as well as diabetes. So even if we are young, and even if we are, our BMI is not that much, but genetically we are susceptible for the development of diabetes South Asian. So we need to look into that. And persistent high blood glucose levels can lead to production of, as I have discussed, reactive oxygen species, leading to DNA damage, promoting to cancer development. Hyperinsulinemia, as I have already talked, that definitely they increase the insulin IGF-1, which is, you can say, an important growth factor for the cellular development. And furthermore, obesity, which is, you can say, चूली दामन का साथ है obesity का diabetes के साथ. We will see in coming slides that also. They also contribute increase of cancer through different kind of, you can say, pro-carcinogenic environment. So understanding these integrate relations between diabetes and cancer underscores the importance of integrated prevention. That's what I am talking, and we all should talk about this because prevention is always easy, and prevention is much more economically beneficial, and definitely this is much more better outcome. And we should all emphasize lifestyle modification, regular screenings, and effective management of blood glucose to mitigate the crashes. All those who are listening to me, if you have a history of diabetes mellitus in your family, you should get your blood sugar checked as early as possible. Uh, So another very common metabolic disorder which we are looking in our population is fatty liver, which is technically known as MSLD. The previous nomenclature was NFLD, that was non-alcoholic fatty liver disease. Now this is known as metabolic dysfunction associated steatotic liver disease. So you can see, you can say, fatty liver is very common nowadays. In fact, any person who has got his tummy protruded, so there the chances were very high chances of, almost half of the person with protruded abdomen, if they undergo some kind of imaging, you will find that there is steatosis. Good thing is that 90% of them, they remains stable. But the bad thing is that this is such, you can say, the numbers are so huge. For example, if you talk about 101 million people of diabetes living in India, even if 40% of them, 40 lakh, so 40 lakh people, they are having steatosis, and even if the 10% of them, they are going to develop SCC in next 2 decades, so we will have 4 lakh people with SCC in coming years. And this is the reality in the developed nation now. the most common cause of hepatocellular carcinoma is MASLD. Rather than previously it was hepatitis C followed by hepatitis B, but now with the advent of, you can say, a very good vaccine, effective vaccine against hepatitis B, and very good screening of, you can say, blood products for hepatitis C, has almost, they are, you can say, very less number of patients of hepatitis B and C nowadays. But and even those who develop hepatitis B and C, we have very good antiretroviral therapy against them, so they can be cured. Now hepatitis B and C patient can be cured, and good thing about that is the treatment is available free of cost by the government across India. Even in our center, we are treating patients of hepatitis B and C free of cost. But MSLE is currently untreatable. We don't have any kind of, you can say, cure for that. And almost ultimately 7% of patients, if they land in hepatocellular carcinoma, that's a very big number. And metabolic dysfunction associated liver disease, um, uh, this is very closely related to type 2 diabetes as well as obesity. And there is, chronic low grade inflammation, as well as, you can say, insulin resistance, which promotes carcinogenesis in these patients. So, as a very high burden of NFLD, and almost more than 13 million people, they have advanced fibrosis. And if you look at the data of global prevalence of NFLD in patients with type 2 diabetes, 55%. So it's very high. And our South Asian region in India, here you can see it is almost 57%, just second to the pancreatic, breast, and endometrial cancer, because these all are arising from the single, you can say, one of the very, you can say, baseline bottom line phenomena is insulin resistance and obesity. And hyperinsulinemia, altered adipokine signaling, dysregulated lipid and glucose metabolism, they lead to abnormal proliferation and impaired apoptosis of cells. So MSLD does not only represent a liver disease, but a systemic manifestation, hepatic manifestation of a systemic disorder with significant oncogenic potentials. And we should definitely look for the importance of early metabolic risk reduction for this phenomena, because it's very important to, you can say, save our population from these kind of things as early as possible. Okay. So now, obesity as a risk factor for cancer, linked to multiple type of cancer, as you can see: breast, liver, gallbladder, ovary, endometrium, colorectal, pancreas, in multiple myeloma, and CA, stomach, as well as esophagus. Definitely, these are, the major, you can say, cancers which are driving the cancer mortality and morbidity across the world. So excess adipose tissue, they promotes chronic inflammation leading to hormonal imbalance. And obesity is, you can say, this is the one of the biggest pandemic of last century. And it is a recognized risk factor for not only diabetes, hypertension, but also of cancer. And the global obesity epidemic has led to increased incidence rate of several cancers, as we have seen. And data indicates 40% of the cancers in the world may be linked to obesity, more so in women. So pathophysiology of obesity and cancer is multifaceted. Adipose tissue, they secrete various inflammatory cytokines and adipokines. And these, they create a pro-inflammatory environment which is conducive to the tumor development. And excess adipose tissue, they also promote insulin resistance, as I am talking again and again about this thing in the diabetes, in the MSLD, and now in obesity. So insulin resistance is, you can say, a common denominator of many of the phenomena in these metabolic disorders. And especially obesity, especially the visceral obesity, meaning thereby the the obesity around abdomen is much more, you can say, dangerous than obesity in other parts of body. The reason for that is that the visceral adipose tissue, they are much more active in terms of secretion of different kind of adipocytokines, which often include the pro-inflammatory cytokines, and ultimately they lead to insulin resistance and increase in hyperinsulinemia, leading to increase in IGF-1. Along with that, there is oxidative distress and all those things. So the hormonal balance associated with obesity lead to changes in cellular growth, and they can lead to problem in the differentiation, ultimately increasing the cancer risk. And it's not only that obesity can lead to, development of cancer, but the prognosis of cancer is quite bad if the patient is obese. They often have more aggressive disease, high recurrence rate, and poor survival rate when compared there to normal weight counterparts. Obesity implicates cancer treatment by increasing the risk of surgical complication and negatively affecting the chemotherapy therapy efficacy. So we need a comprehensive studies focused on, you can say, weight management, physical activity, dietary changes. Especially obesity is now very common in our school-going children, so we need to take care of them. Childhood obesity is much more difficult to treat than the adult obesity. Child Obese child invariably turns into an obese adult. So it's very important to take these precautions early in the course of the development of obesity. We should watch the way I always, whenever I get a chance, I talk about this, the way we take care of their, their health, their education. So we should also take care of their weight. If it is increasing, then definitely we need to look into it. In the beginning, it's quite easy to handle, but once if the patient has developed obesity, then it's a self-perpetuating phenomenon, very difficult to treat. So everyone, men, women, boys and girls who are listening to me, you should watch your weight, you should watch your belly, and you should measure your waist circumference. And if it is more, you should take care of them at this moment only and start right now, because it becomes very difficult once you are having full-fledged obesity and its related complications. So what are the pathophysiological mechanisms? Here you can see the metabolic anomalies leading to different kind of things, which I have already discussed, leading to altered metabolic state, leading to problems in the DNA, you can say repairing, leading to more instability, oxidative stress, epigenetic changes, and all of them, they, you can say, promote carcinogenesis in some form or other way by promoting immune evasion and all those kind of things. Another thing where metabolomics can play a very important role, early identification of any disease, especially cancer, is very important, because if you are catching them early, your half job is done, because it's very easy to remove the cancerous part. It's the chemotherapy or the immunotherapy, whatever you are giving, they are much more effective in the initial stages. But if the same malignancy has, progressed to different parts of body, meaning thereby they have metastasized, then definitely you can say the risk is, the prognosis is not that good. So metabolic markers, they offer promise for early diagnosis and also for risk stratification and prevention. And they can identify individuals which were primed for malignancy before genetic or imaging signs can appear. They serve as early warning signals for timely therapeutic interventions, and they enable restoration of metabolic balance to halt the cancer progression, and they highlight the translational potential of metabolism-based diagnostics and link to metabolic dysfunction with oncogenic signaling pathways. Just a small example of ALF10 and metabolic pathways is cancer prevention. You can see the hepatic transcription regulators connect lipid metabolism with cancer signaling. This ALF10 protects cancer against steatohepatitis via HNF4 alpha dependent lipid and bile acid pathways. They bridge the metabolic stress, inflammation, and hepatocarcinogenesis, which we were talking about. So we have got some practical example of that thing. So targeting a metabolic dysregulation offers preventive and therapeutic strategy. And early metabolic shifts may reveal novel tumor initiating pathways. And if you identify that, then definitely if we identify some kind of pathophysiology, then it's very easy. Not very easy, but at least we have something that we can target. So integration of the metabolomics into the genomics and proteomics enables precision prevention as well as precision treatment, which is you can say talk of the town nowadays, that is precision. And this is the same thing which I have talked. So metabolic dysregulation, inflammatory state, and other factors, they are the common pathological mechanism between cancer and metabolic disorders, which we have seen. And these are different kind of pathway which we have seen previously in the obesity, in the diabetes, that this phosphoinsitol kinase 3, AKT, mTOR pathway, MAP kinase pathway, IgF bioactivity, and Warburg effect, which where we have seen that there is, I mean, you can say that there is a basic change the way a cell behaves, especially with the bioenergetics, which leads to, you can say, their increased chances of survival, and less chances of apoptosis, and more chances of, you can say, spreading across the body. So these are the things which can be explained with the help of hyperinsulinemia. Along with that, there is inflammatory and metabolic diseases, for example, age, enrage, excess, on this advanced glycation and products. They are very, you can say, basic, I mean, pathophysiological defect which happens in patients with diabetes because of increase in the level of glucose. There is increase in the glycation of different kind of proteins, and there is a receptor for these advanced glycation end products at the different cellular level. So this age and rage combination leads to epithelial transformation, tumor resistance, and epithelial dysfunction, which is responsible for multiple, you can say, complications of diabetes, including diabetic neuropathy, diabetic retinopathy, diabetic, you can say, cardiovascular disease in diabetes, heart failure. So they are also implicated in development of tumors. And definitely we know that digestate pathway is one of the very important growth pathway. And if there is inflammatory cytokines, then there can be oncogenic gene transcriptions, nuclear factor kappa beta activations, which is, you can say, central to the development of inflammation. All these lead to angiogenesis and tumor progression. And metabolic reprogramming, both disease involve altered glucose, amino acids, and lipid metabolism. And this we have discussed already, that high risk for, you can say, pancreatic cancer, liver cancer, and colorectal cancer. And there is moderate risk for development of prostate, gallbladder, and obesity related, definitely postmenopausal development of breast cancer, endometrial cancer, cervical cancer. So these things are, you can say, quite, obesity and diabetes, they do play a role in development of these cancers in some way or another way. You can say they are basically complementary to each other rather than to compete with each other. They complement with each other. Now coming down to the therapies which we have at this moment. GLP-1 receptor agonist. This is something now everyone is talking about them, because more they are, you can say, one of the most effective agents against the obesity, not only diabetes but also obesity. And almost 90% of the works of bariatric surgeons, they are done, because almost 30% weight loss can be achieved with the help of these GLP-1 receptor agonist or dual agonist against GLP-1 and GIP. And we have triple agonist, GLP-1, GIP, as well as glucagon. So they are in different stages of development, but the GLP-1 and GLP plus GIP, which is known as tirzepatide, GLP-1 receptor is semaglutide, which is available, and GLP plus GIP is tirzepatide. Both of them, they are now available in India, but quite costly therapy, at least 15,000 per month. But their patent will be off in March 26 next year. So a lot of, you can say, generic brand will be available at a very nominal cost of, you can say, around 2,500 to 3,000 per month. So they do have a very good efficacy against reduction of HbA1c, which is, you can say, one of the parameters which we target while treating a patient of type 2 diabetes mellitus. And weight loss, this is 3 to 8 kg, but more than 20 kg weight loss has now been observed, especially those who are having, you can say, morbid obesity. And very good effect on cardiovascular events. They are cardioprotective also, especially the atherosclerotic cardiovascular disease. They are very much, you can say, effective in preventing them. preventing the adverse outcome in patients of atherosclerotic cardiovascular disease. And this their preventive effect on this cardiovascular outcome is independent of their glycemic efficacy, meaning thereby if you lower down the glucose by any other medication, and if you lower down the glucose with the help of GLP receptor agonist, and you keep both the groups on the same level of glycemic control, the cardiovascular outcomes is much more beneficial in patients who has got who will receive GLP-1 agonist when compared with the other kind of therapy. And because they are leading to weight loss, they are leading to, you can say, and there is decline in the pro-inflammatory state, there is decline in the insulin resistance, insulin sensitivity increases, and there is increase in the GLP-1 signaling on the immune cells also. So these things may have anti-cancer mechanism. And there are certain clinical evidence that from the observational studies that there is almost 40% reduction in obesity-related cancer incidence if you use GLP-1 receptor agonist along with metformin. And definitely additional benefits, as I've already talked, they are there. Extra glycemic benefits, which we talk about them in our own terminology. Another is SGLT2 inhibitors. We have done some work with the SGLT2 inhibitors in carcinogenesis. We have got a very good animal data. Uh, mm, Actually, I was not able to calculate that data because because of time constraint, but that work has been done in our university only, along with our basic science colleagues, where you can see that SGLT2 inhibitors, they are, you can say, that is sodium glucose co-transporter 2, which is present inside the kidney, the renal tubules, the proximal tubules. How they have antitumor effects because they block the glucose resorption from kidney. There is less glucose supply to the tumor cells, and almost 70 grams of glucose can be excreted by the SGLT2 inhibitors. So cancer effects, they include GLUT1 expression. They will definitely have an effect on Warburg pathway, and they increases the AMP kinase activation, which is, you can say, one of the major, you can say, healing pathway in our body. And because of this energy crisis, because of decrease in glucose, there is decrease in ATP production, so that can increase the tumor cell apoptosis and will reduce to proliferation also, and that may also increase the chemosensitivity as well as radiosensitivity to different kind of therapies which can be given. And definitely there is benefits like weight loss, HbA1c, and they are very good drugs for, you can say, heart failure. And the management of heart failure has changed in last. Major pillars is this SGLT2 inhibitors. So they have got very good effect on heart failure as well as in the patients who has got renal disease. Not only patients who has got renal complication because of diabetes, but other kind of kidney disease patient, they are also being benefited with the help of SGLT2 inhibitors. So what if we combine both of them? So we do expect synergistic effect because they have got complementary kind of mechanism, because both of them, they reduce hyperinsulinemia, weight loss via different kind of pathways, and metabolic advantage will be superior HbA1c reduction and more sustained weight loss in long and short term. And definitely you can see that the cardiovascular benefits are much more with the help of GLP-1 analogs. If the patient has got atherosclerotic cardiovascular disease, and if the patient has got heart failure, then the benefits are more in patients who are receiving SGLT2 inhibitors. And definitely in renal patients of diabetic kidney disease or renal failure because of any cause, the SGLT2 inhibitors like dapagliflozin or empagliflozin, they are very effective in, you can say, reducing the risk of progression of the diabetic kidney disease. And definitely, so they are complementary to each other at different levels, and definitely they have potential for targeting multiple cancer promoting pathways if we combine both of them, especially if the patient is obese and have diabetes. Definitely metformin is one of the wonderful drugs. Even after 60 to 70 years, it remains the first line therapy in most of the uncomplicated patients of type 2 diabetes mellitus, because of reduction in hyperinsulinemia, reduced mitogenic effects and cancer risk. And also there is reduction, there is AMPK activation, so there is reduction in this mTOR signaling pathway, which has anti-proliferative and pro-apoptotic effect. And definitely it is also useful in weight management as it promotes modest weight loss. And definitely it has one of the drugs which has got very good cardiovascular safety as well as benefit. So even we have tried a project of metformin in cancer HXCA, but we were not able to, you can say, convince the funding agency. But definitely we are trying to do something with metformin also in our university for cancer. So what is the final thing that now what we need to is integration and multi-specialty, multimodality kind of approach is needed in every disease, including cancers. So we need to have tailored therapy selection based on not only on glycemic control, but also on the weight, the cancer risk, the comorbidities in the form of heart failure, CKD, ASCVD. And we need to have regular monitoring to develop, to see the adverse effect of these drugs, and also to see the beneficial effects of these drugs. So, what are the things which can change in clinical practice? Is that we should select anti-diabetic therapy considering the cancer prevention benefit also in long run. But for that, we need more, studies, as you have shown that metformin plus GLP reduces the risk of cancer incidence by 40%. But we need more data so that we should give the benefit of evidence-based medicine rather than based on our own judgment or our own thinking, because the era of evidence-based medicine is over. Now we should talk about the evidence-based medicine. Wherever the evidence is there, we should share with our patient, and we should try to give the maximum benefit, which is based on some logical reasoning rather than our own, you can say, comprehension of anything. And first and second and third line drug can be decided according to the patient's condition, patient's affordability, patient's, you can say, the comorbid conditions. And early intervention is definitely, we should try not only from the cancer perspective, but also from the perspective of development of diabetic complications. So there are multiple research opportunities in future. Definitely validate cancer incidence reduction with dual therapy. We need more mechanistic research how linking the diabetes, obesity, MSLD, and cancer, how actually they are converging to one thing. And definitely we need more biomarkers, more less kind of, you can say, less invasive, or you can say non-invasive or liquid biopsy kind of things, by which we can diagnose, we can characterize, we can prognosticate, we can provide the risk stratification based on the metabolic markers. And definitely therapeutic combination based on these things should be provided to our patients. So just to summarize my talk, metabolic disorders and cancer, they share the fundamental pathophysiological pathways, and we need to have strategic use of anti-diabetic agents, which offers dual benefit of optimal glycemic control and reduces the cancer incidence and mortality, have cardiovascular protection as well as benefits in the kidney, and they should improve the quality of life also. That is also very important. अगर जिंदा है तो जिंदा नजर आना भी जरूरी है. What happened? So that's all. Thank you very much for your patient listening. If there are any questions, then definitely I am happy to take those. I think there are no questions in chat box. Uh,
I think I'll read that. Yeah, sorry, go ahead.
So we have got four kind of parameters. One is fasting, another is postprandial, third is random, and fourth is HbA1c. So if any one of the tests is above, you can say, above the normal range, then definitely we should start the treatment. And treatment does not only include the medication. The first line of treatment still remains diet and lifestyle modification. So any patient who has got HbA1c of more than, 6.5, or fasting of more than 126, or postprandial or random blood glucose of more than 200, they should be started on treatment. And treatment may or may not include medication, because diet and lifestyle modification is as good as any kind of medication. So we tell our patient that this is the first and best and safest medication, and also the cheapest one. So any one of them, if it is abnormal, then we should go ahead with the treatment. You should consult your doctor and start the things as early as possible. And if you talk about the sensitivity, which is the most sensitive test, is, you can say, the postprandial glucose is the most sensitive, but you can say many times they are they are not reproducible. So HbA1c is also a good test in that scenario, that it gives us an integrated measurement, and along with that, there is no need for fasting to do an HbA1c. It can be done at any time. So every test has its own benefits, and you can say risk.
Can I take one question?
Oh, why not? I will be happy.
Okay.
If I'm not able to answer, then you have to give the answer also.
No, it's a genuine question.
Yeah.
I was just thinking about, there are some known mutations or which are known to cause cancer. Is
Okay.
there a way that we can change something at the metabolic level, whether through drugs or whether through lifestyle changes, that can mitigate the effect of that mutation or the effect of that gene? Because that will be something that will be crucial to understand that these omics are playing together, or DNA is the master regulator.
Definitely. This is something I think where the focus is now going on, that we should identify different kind of mutations, especially those who where the things are common. For example, if I'm talking about PI3 kinase or mTOR or MAP kinase, if these kind of things are activated in any cancer, then definitely, because these are the things which is also activated in different kind of metabolic disorders. Repurposing of drug is also very important. So definitely, maybe they don't have the curative effect, but at least they will have some kind of beneficial effect, in terms of that they might increase the sensitivity of the kind of traditional chemotherapy or any kind of immunotherapy which we are giving to the patient. And definitely they might increase the sensitivity, they might increase the cure rates, they might increase the, decrease the relapse rate. So all those things, I think we should, evaluate, because with the advent of these generic GLP-1 analogs, I think we should talk more about these therapies along with the conventional therapies which is available currently.
Thank you. I think there's one more question.
So what? Basically, what we are talking about right now is not the risk reduction. It's basically, clinical improvement, because risk reduction is something which is in the primary prevention. We don't have data, that much solid and robust data for primary prevention. But if the patient has already heart failure, or if the patient has already renal failure, in that situation, definitely the risk of hospital readmission is almost reduced by 40 to 50%. Risk of mortality due to heart failure or any heart condition is reduced by, you can say, 30%. So they are, you can say, a very big boon. And one of the very important thing is that they are very, because of the patent is off, now you can get a SGLT2 inhibitor at the cost of, 6-7 rupees also per day therapy. And in patients with heart failure, there is also a issue of, you can say, low BP, hypertension. So they do not produce hypertension. They do not cause hyperkalemia in patients of renal failure. So definitely they are, you can say, a very good agent to reduce the risk of death, as well as the reduce the risk of hospitalization in patients of heart failure. And they are also very effective in reducing the proteinuria, which is one of the, you can say, hallmark of diabetic kidney disease. And they also reduce the risk of progression of diabetic kidney disease. For example, if the GFR declined by 5 to 10% per year, then definitely this decline is reduced to 1 to 2% per year. And the patients are, they are being prevented from the landing into the end stage renal failure. If the patient has got heart failure and if the patient has kidney disease, whether the patient is diabetic or non diabetic. Okay, thank
Actually, it
you.
will be a long break for the lunch. It will be 1 to 2, so we'll reconvene at 2. Is that right?
Okay, thank you everyone. Bye bye.
Thank you, Professor Hamid. I personally thank you for enriching the session and this entire course with your insightful lecture. Thank you.
Okay, bye.
Good afternoon, everyone. Can you hear me?
Good afternoon, sir.
Very good afternoon, Swati. Can I access, please? Thank you,
Yes, sir.
Khadija.
Just a moment. So people are still joining. We
Yeah,
can wait for 1 or 2 more minutes, if that is okay.
yeah. It says I'm not allowed to share my screen, so
I have made you co-host. You can
okay.
check it
Thank
out.
you. I'm still getting the same message. You're not allowed to share your screen.
Okay.
Working now. Thank you.
Thank you, sir.
Swati, could you please confirm you can see the slide as full screen as well as the pointer in red?
Yes, sir. It's working. I
Perfect.
can
Thank you
see.
very much.
Okay. Welcome back, everyone. I hope you enjoyed the break. Now we are going to proceed with our next lecture by Dr. Mohammad Asim on the topic the drivers of cancer. We warmly welcome him again and look forward to an engaging and thought provoking talk. Sir, over to you.
Thank you very much. Uh, okay. Do we need to wait? I'm seeing only 14 of people have joined, 15 now. So shall we wait a couple of more minutes? Or
Yes, sir. A few minutes would be nice.
this is being recorded, right?
Yes, sir.
Okay. Uh,
Meanwhile, if anyone has any question or
yes, yes,
um,
sure. Please
okay,
feel free to in the
yes, sir.
chat box. So Khadija has Khalida, I'm sorry. She has joined, and so has Miss Shiza Siddiqui, it seems. Very good afternoon, Shiza. How are you? Brilliant. Thank you. Okay. So shall we crack on?
Yes, sir.
I think there's someone saying something, or I was hearing an echo.
So is it working fine now?
Yeah.
I think it was
You
some background noise from my end. So
okay?
sorry.
All right. Okay. So this is day 3 of this course, and I'm conscious that some of you are, in fact, BSc students, as we saw yesterday, and we have not a big crowd today as we saw yesterday. So we will start, and in case there will be questions, please feel free to keep posting them in the chat box. I'll try periodically to have a look while I'm teaching and try to answer them. If not, then we can take the questions afterwards. Does that make sense?
Yes, sir.
Good. Okay, thank you. So, what we are going to learn today is about some genes. So, in subsequent lectures, we will know what is the effect of DNA damage on gene functions. We saw in the last two lectures, one was the damage of the DNA that occurs, and yesterday we saw what are the pathways which are there to repair the defects in the DNA. But we also learned why some cells, cancer cells, become vulnerable to drugs because they cannot really repair their DNA efficiently. So today we are going to see what sort of genes are mutated by these changes, by these agents, by natural processes, by spontaneous changes happening within a cell. So, what are these genes? We're going to talk about these genes called driver genes or oncogenes today. We will see what they are. And we will learn that 95%, or shall I say over 99% of the treatments of cancer we have currently is based on targeting some sort of targets. Chemotherapy aside, which targets the DNA replication, DNA integrity, most of the targeted treatment of cancer actually are targeting a driver, which is an oncogene, as we will learn in the lecture today. So, then what we are going to discuss in the next hour is these genes called tumor suppressors. I've been calling them good genes in the past 2 days, and just to make sure that I not only have faculty joining this call, but also students very early in their career ladder. So please forgive me if there is something which looks like super basic biology to you. And in addition to discussing tumor suppressor genes, we are also going to discuss how some viruses can cause cancer today. So there are genes which are known to be involved in cancer. If you take DNA out, sequence it in from patients, you will find that there are mutations, which we discussed yesterday, day before yesterday, actually, what is the definition of mutations. So the genes can be broadly classified based on their role in cancer into being proto-oncogenes, which are, should I say, ticking time bombs in a cell that can cause cancer. Then we have tumor suppressor genes. These are the good genes, which I've been mentioning in the past few lectures. And a lot, but not the least, we've spoken yesterday about genome maintenance genes. We discussed these genes, which are there to repair any defects, any threats that come to the DNA. They can actually deal with that. They can allow DNA to be repaired so that the DNA damage does not really manifest itself into a full-blown mutation. We discussed that yesterday. Any question on what we covered yesterday or so far? This is important because this will set the foundation stone for what we are going to discuss today. So if you have any confusions, please feel free to ask me any questions now, or as I said, I'm more than happy to answer any questions through the chat box. Okay, let's move on. So what happens in cancer is you see these genes, we call them proto-oncogenes and oncogenes. I'm sure some of you might know them already. Does anyone know what do they do? Why are they there? If nature knew that these genes actually can cause cancer, why would nature give us these genes? And the rationale for this is they are needed for development. They are needed for normal growth. They are needed for proliferation. They're needed for healing, especially healing of wound. Have a look at this child here. And a few decades later, it becomes this. So this is none other than myself as my childhood photo. You can see I needed those genes, proto-oncogenes, in order to grow and become what I am now. So you need these genes in order for growth. So you can see this as a necessary evil. We do need these genes in order to grow, proliferate, develop, um, progress, heal. So we have to have proto-oncogenes. The key difference is that proto-oncogenes can actually be mutated by pathways which we discussed yesterday and day before yesterday. And this mutation can cause them to become oncogene. Now, there is no 1 plus 1 equals 2 formula here. Uh, there may be different kind of mutations taking place in different part of the protein, which can cause these gain of function mutation. That is a very peculiar term. Please remember that, because there will be some questions. Those of you who are attending the exam and wishing to get a certificate, is that there is a gain of function. So proto-oncogenes have their own inherent function. But on top of this, they get an anomalous and abnormal function, which causes them to not be regulated. So although proto-oncogenes can be switched on and off, as you can see from this alternator button used in some batteries, some inverters, you can switch on and off the function, just like in proto-oncogenes, when they are needed to take this person from here to here. Some of them may get switched on, others may not be switched on. But what happens as a result of mutation, this barrier, this regulation is abolished. And when this regulation is abolished, you can actually have a system where there is no off button left. So the genes are always on, meaning they're sending signals to grow, which is a bad news. And that is why nature cannot avoid existence of proto-oncogenes in our cells to grow, proliferate, heal, but that is a necessary evil. What is not desired is to have the mutations. And as we discussed day before yesterday, uh, we have some part to play where we can suppress the rate of mutations we get in cells through lifestyle changes. But then there are sadly parts which we cannot control, and that can lead to transformation of proto-oncogenes into oncogenes, which become switched on. And as I said, and we will discuss what is happening here, what type of mutations, what type of changes are happening in the DNA of these genes, causing them to become oncogenes. So when I said to you the gain of function mutations, which give them an abnormal function, sometimes at the expense of their regular function. So sometimes they may not be able to carry out their regular function, but 90% of the time, their primary function is over amplified. So now they're able to carry out their primary function more than what was desired. On top of this, they do not respond to any signals which were meant to block their function. In other words, they become autonomous. So what type of mutations you might find is that leads to the gain of function is one of the primary mutations is the point mutation. And we discussed this on day one. What are the point mutations? What can they lead to in the DNA and the alternative products that they can actually make? Then what we also discussed was sorry, not discuss, but we can also have chromosomal translocations where two genes on two totally unrelated chromosomes can come together as a result of the arms of different chromosomes shifting hands, and that can lead to formation of hybrid genes. And last but not the least, amplification. So sometimes, so in a diploid cell, usually you will have two copies of genes, but what can also happen is that a particular gene is amplified multiple times. In cancer, we know some of the driver oncogenes, and I'll give you an example today, where we see sometimes a copy of the gene is not as such mutated, meaning there is no single base change, for example, or two genes fusing together, but amplification, which overall would lead to change in the sequence of the genome on that particular chromosome. But because you have lots of genes for this, lots of copies of the same gene, they're going to make a lot of the same product which is not desired. And I'll give you an example today. uh, as I mentioned in cancer, what happens with this. So have a look here. This is one of the classic textbook example of normal proto-oncogenes converting themselves into an oncogene as a result of mutation. And you can see here what happens. This is plasma membrane. This cartoon shows, and there is a receptor which is anchored within the plasma membrane. This is a protein. I'm not going to go into the details what sort of amino acids you will find in this region or this region, but basically this part of the receptor is the receptor binding part to the growth factors. So epithelial cells, which we discussed from majority of human cancers, they need to grow. And when they need to grow, the signal comes from another cell type. Sometimes they're called growth factors. And their binding would activate the receptor. And as a result of this, the receptor dimerizes, you can see, by binding of the growth factor, and then the internal tyrosine kinase domain, in this case, becomes active, causing signal transduction pathways to become active and allowing these cells to divide, progress into cell cycle sometimes, of course. So what happens in some mutations which happen in this scenario is that the receptor may not need the growth factors at all. In other words, they become self-sufficient. So they've lost a switch on, which is switch on will be the condition when growth factors are bound. Switch off will be when the growth factors are not bound. This is regular switch on, switch off, switch on, switch off condition. Right? But what happens as a result of mutation? You can see there are mutations in some of the domains as shown by this red dots. And as a result of mutations, you can see still the receptor is able to dimerize. Here it was not dimerizing without the binding of the growth factor. So the receptor needed, in order to become dimerized and activated, it needed the growth factor. And in this case here, there is no requirement for it to have growth factor. Just because it has some mutations, it activates those receptor dimerization. And as a result, the signal transduction pathway that allows cells to grow, divide, proliferate, they become always active. Another example would be that there are some stop codons created at the level of DNA due to the results of mutations. And what happens here, because of these, you may actually altogether lose a part, a domain of the protein. In this case, the extracellular domain of the receptor is gone. Why is it gone? Because there are changes at the level of DNA mostly, and these changes would introduce a new stop codon before the conventional stop codon, meaning the protein is going to become shortened. This protein, unfortunately, will still be active, but it will not require a growth factor to activate it. So it will always be firing and sending signals for the cell to grow, which is of course a bad news. So both scenarios, you have switch on, switch off, switch on, switch off. But in this case, you always have this situation or this situation. They're always switched on. And that is a bad news because it is going to allow these cells to have a sustained growth promoting signal, which can actually allow these cells to start to take the first step towards growing uncontrollably. They may not form any cancer at this time. Remember what we discussed yesterday? Cancer is a multifactorial slow process, and it requires a number of subsequent mutations after the first mutations in order to cross all these barriers to become full-blown cancer, which is able to circulate in blood, sit in new organs and grow. So this is one of the first steps go on a cancer pathway. The example will be this mutation here, the red dot I was mentioning to you. If it is a HER2 gene, and I'll come to this gene in a minute, what is known is that there is a mutation which causes valine to glutamine point mutation. And you can see this is a HER2 receptor extracellular domain, the intracellular domain, and in a normal wild type form, there is a valine here. And what happens when the signal comes? It requires the activation through the binding of the HER2 ligand. What happens sometimes is that you can have this mutation which causes a valine to become glutamine, and this point mutation is found in breast cancer. I'll come to that in a minute as well. And as a result, what happens is that this glutamine, when valine is replaced by glutamine in this part of the receptor dimerizes and sends the growth promoting signal all the time. And that is therefore an oncogenic mutation. And this is represented by valine, so the three-letter code for amino acid val, and then an arrow and glutamine, which is GLN. And that is something that doctors, pathologists would look at. There is HER2 overexpression. And on top of this, what is happening? There are mutations. So HER2 gene itself is amplified in breast cancer. And sometimes what can happen is this can unnecessarily, these small changes can unnecessarily activate, stabilize a protein. Normally you have proteins being formed and dying. They're being formed and they are then degraded. They're not needed, they go away when they're not needed. By having some of these mutations, they can actually escape their degradation. So they don't really die anymore. They don't and get degraded, if you understand what I mean. So what happens also in this gene is, I can tell you this is EGF receptor, epidermal growth factor. HER2, by the way, is nothing but also an epidermal growth factor receptor. And what happened here is that as a result of mutation, you can see that the extracellular domain of this EGF receptor is gone. I was showing you this similar thing on a previous slide. And as a result of this, what happens? This was the extracellular domain, which was keeping these two dimers to form and become active. By losing this extracellular domain, the protein dimerizes and starts sending, relaying a growth promoting signal as a result of it. So it's a shortened copy of the receptor. Normally, what should happen to a cell? If it produces a protein which is mutated protein, cell would degrade it, right? Other times, what will happen is what you're seeing on your screen, that these proteins still are not able, they escape the degradation, they escape being killed by a cell, because cell knows that this is not the protein which I desire to have. But in this case, what happens, it is still becoming active and leading to the continuous, unstoppable growth of cancer cells. And if I show you some real life example, have a look here. This is an immunohistochemistry slide from a woman with breast cancer on the right. This is how it looks in normal cases. So normal breast epithelial cells, you can see here. If you do immunohistochemistry, which you learned yesterday, what happens is there will be some expression of HER2. Yes, because we need HER2 in order for cells to grow. Epithelial cells are one of the fastest growing cells in human body. So they will need these epithelial growth factor receptor signaling, right? They need it all the time. They are some of the fastest growing cells. As I said, our own gut, the entire gut is replaced by some estimates. Within 2 or 3 years, the entire gut, we have a new gut, basically. Do we see it? No, because the cells are changing, old cells dying, new cells taking their place. Dr. Aziz can tell you about this in more precise details, but actually the whole gut is replaced. So the division is really, really quick because they're sitting at the forefront of dealing with things, external agents, UV light inside our gut. We eat all kind of junk foods, and as a result, the body knows that there are more chances of having a damage, and a damage that can actually lead to mutations. So what happens here is that the HER2 is expressed in those cells as well, normal breast epithelial cells. Have a look here what is happening. You have an enormous expression by these epithelial cells, the glandular epithelial cells of the breast cancer. They're expressing, you can see from the signal, the dark brown color, there is substantially higher expression of HER2 in these cases, uh, women. So their cancer is driven by HER2. And as much as over 30% of the women will have uh, their tumors being driven by HER2 kinase overexpression. So it is being overexpressed, meaning there are more protein which is being formed. There is more protein copies of HER2 in these cells, and they are sending a sustained proliferative growth division signal to these cells. And that makes actually HER2 an excellent target. Why? Because this is overexpressed, and this is overexpressed on the surface. So you can actually devise an antibody, a monoclonal antibody, as a therapeutics. So drugs don't have to be, as you know, always small molecules, chemical entities. They can be biologics as well. And an excellent example, which has been very successful in treating patients who have their tumors being driven by HER2. You can see here, they respond very well to this drug called trastuzumab, or Herceptin as we call it. And Herceptin is a very successful drug, but only in a subset of patients. How big is the subset? 1/3 of the patients. So when a patient comes to the clinic, doctor will take the biopsies, and among other things, looking for other oncogenic drivers. They will also look for drivers, for example, HER2, because a lot of women express it. And if the tumor is positive for this, they see an over representation of HER2 expression in their tumors through this IHC technique, then those women may be prescribed Herceptin. And that may actually lead to treatment getting successful. If it is given without any knowledge to a woman whether their tumor is driven by HER2 or not, the treatment is not going to work, especially if there is no HER2 overexpression, uh, which is a bad news. And that's why this is what we call personalized medicine. You need to see the underlying changes in a cancer cell, what these changes are. And on top of this, can you target those changes through a drug? This drug can be an antibody as shown on your screen. It can be a small molecule. And in both cases, what you can do is devise treatment based on the expression. This is what we call personalized medicine. The problem, unfortunately, we face is that in this case, a lot of women express HER2. This is not the case for many cancers, as I'll come to this in a minute. You would see that there are oncogenes which are expressed in 5% tumors, in 2% tumors, in 3% tumors, and therefore it's very difficult to see which one you want to target. As Dr. Aziz yesterday alluded to, it is a moving target, basically. It can change, cancer can change its reliance on some drivers at some point in their life. Later on, they may actually shift, and because they're highly plastic, the tumors are show very highly plasticity. We will see tomorrow as well, metabolic plasticity. But because of this, it is very hard to treat, unfortunately, a disease like cancer. So what is the impact of the treatment? Women have the tumor driven by HER2. You can actually treat them with drugs like this. What about other women, 2/3 of women? So as I mentioned, 1/3 of the women, up to 1/3 of them will have a HER2 overexpression. What about the rest? They have the tumor driven by something called nuclear receptors, which we will come to in a minute. And these are called nuclear hormone receptors. But I want you to have a look at this slide showing survival of women with HER2, where the HER2 is present or not. And have a look what is happening here. You see that in chart A, this is called what we call as a survival curve or Kaplan-Meier plots. And what you can see, the this is percentage survival on your y-axis, and this is survival in months. So you can see purple, the survival is very, very poor. If you have this particular marker, only 20% patients will be surviving after 4 years, as you can see. So let's have a look what is purple. Purple is basically HER2 negative tumors. They don't have HER2. Now you would say, hang on a minute, you were saying that HER2 is a driver. They don't have HER2. So why are they dying so enormously that only 20% surviving compared to the green one, where actually more than 50% surviving when they have HER2? I'll tell you in a minute. Have a look what is happening here with HOR, which is hormone receptor. So hormone receptors are the other uh, substantial category which drive breast cancer. And which hormone receptor? Estrogen receptor is the one that drives, and also the progesterone receptor. They are the ones that drive breast cancer. So what is happening? What is the group that is doing the worst? The group of patients. The patients who succumb to their disease are the ones who do not have HER2, who do not have hormone receptor. So sadly, these are the patients where we don't know the target. Now that slide tells you how important it is to identify targets, a drug target, to save lives of patients. You can see here HER2 negative and two hormone receptors missing. We call it triple negative breast cancer. Why triple negative? Because they don't have estrogen receptor, they do not have progesterone receptor, and they do not have HER2 receptor. So this is something which we don't understand very well. They don't have any conventional targets which we can block using drugs. On the other side of the spectrum, we have the tumors in green, and they express HER2 in women, and they also express hormone receptor. We can target HER2 by Herceptin-like drugs I showed you on the previous screen, but we can also target the hormone receptors using, for example, estrogen receptor can be targeted by drugs like tamoxifen. You can aromatase inhibitors as well, and you can see here if we know what drugs to give these patients whose tumors are driven by these drivers, the outcome of the treatment is actually very well. You can see here after 4 years, almost 50% of the women surviving. Sadly, where the tumors don't express these, there is, you can see the survival is very bleak. Only 20% women are surviving. And if you see other charts, they actually tell you a pretty similar story here. This is breast cancer specific survival, and you can see this is the overall survival, and that tells you how bad the graphs see how bad is it to have a metastatic disease. You can see the group doing the worst is the one where the cancer has gone into the brain. It has metastasized and it is in the brain. And you can see the patients who have the cancer going into liver, lung, bone, they're still doing better, shall I say, compared to those where the cancer has gone into brain. And this is largely because we can actually perform surgeries. Not only that, there are agents which you can give to strengthen bone, again, strength back, like bisphosphonates and so on, that will allow to mitigate some of the side effects of metastasis. But eventually, all of these patients sadly will succumb to their disease. Okay. So sometimes what can also happen, I'm telling you, I was telling you in the previous slide that you can lose these domains which will allow the cells to become active. Also, you can have the mutations I was showing in red dots. But there is something else which can also happen. The cell itself might gain the ability to make their own growth factor. Previously, in the previous similar chart I showed you, the signal for the growth factor was coming from a separate cell being produced elsewhere and being used here. But now these cells have become autonomous. They do not really need growth factor from a different cell to allow them to grow. They become autonomous. So you can see here in this uh, staining, this is called immunocytochemistry, or immunofluorescence. Actually, you can see here that there are cells within this tumor stained with antibodies that give fluorescent colors. So for example, this is the same tumor. You can see here it is expressing epidermal growth factor receptor, something like that. Yeah. And what is also happening, this very same cells are actually, if you do the staining for epidermal growth factor, the very same cells are also expressing the epidermal growth factor. So they don't really need the signal coming from a different cell type. So you might find sometimes that, oh, this cancer patient, if we do the sequencing, it looks like the epidermal growth factor protein looks normal, the gene is normal, there is nothing wrong. But have you looked at whether these cells are starting to make their own epidermal growth factor? So there may not be mutation as such in the receptor, but this time the cell gains the ability to create, to make their own growth factor, allowing it to have a sustained proliferation, which can actually lead to cancer. Okay, so what is happening here is I was mentioning to you that herceptin targets the HER2 receptor, and then I introduce you to nuclear hormone receptors. What are they? Basically, nuclear hormone receptors are nothing but some receptors which are not present in the plasma membrane. If you close your eyes and as a biologist and mostly think about, oh, receptors, they're sitting on the cell surface. Many students do these kind of mistakes. I know full well. Basically, these receptors are receptors in this case because they receive a signal from androgens, which are testosterone, and estrogen, which are female sex hormones. And the receptor itself is not on cell surface. Think about that, why they're not on cell surface. Primary reason is the testosterone, estrogen, they are both hydrophobic. They can simply traverse through the plasma membrane. They don't need any gates, any channels, any receptor to be present to receive their signal on a cell. They simply come inside the cytosol. That's exactly where the receptor is. So in case of androgen receptor, where my lab has been studying their role in prostate cancer, the androgen receptor can cause prostate cancer. You asked me where the prostate gland is. It is situated underneath the bladder surrounding the urethra. So some older males will have problems passing urine. They'd feel that their bladder is full. And exactly what is happening is sometimes the prostate is growing uncontrollably. Is it cancer all the time? Not really. Many times it is a benign non-cancer condition, which is age-related. But sometimes you can have a diagnosis for prostate cancer as well. So we call this androgen receptor. What are the chances if you're a female to have prostate cancer? Exactly zero, because females don't have a prostate gland. As opposed to breast cancer, where males and females both can get breast cancers. Of course, the chances of breast cancers are much higher if you're a female. So let's spend next 10 minutes on studying the role of these nuclear receptors as oncogenes and as drivers of the disease. So have a look at this slide. This is a PET-CT scan of a patient. And you can see this is primary cancer, meaning the primary cancer simply means the cancer resides in the primary organ, which is the prostate gland in this case. And you can see here that if you take out the biopsy is done to stage the tumor to know, is it just a start of tumor or is it something which is really at the stage 3, stage 4, meaning very advanced stage, where the treatment will become difficult, right? So if you take out these prostate glands and stain the epithelial cells, of course all the tissue will be stained, and do the staining, you might find an increased staining. As you can see here in brown color, these cells are making more androgen receptor. An androgen receptor is known to drive prostate cancer. Patients who don't have the androgens, they actually never develop prostate cancer. Have a look what happens when this disease will progress. So at the moment you're seeing in this patient, there are some vertebra in the spinal cord, sorry, vertebral column. You can see the presence of tumor as shown by this PET CT. I'll tell you tomorrow why, how this works, PET CT scan. Have a look in the meantime what is happening here. So if the disease progresses on, despite being on a drug, you can see here the black staining, the imaging, that the cancer actually unfortunately has gone into bones. If you take out the bone sample, so you would ask me who would give the bone sample to if you're alive and you want to get give your bone sample. Basically, I have a friend who is an orthopedician, and he sees a lot of prostate cancer patients. So 15 years ago, I asked him naively a question: how come you see prostate cancer patients? You're a bone doctor, you're an orthopedician. And his answer was simple: because it causes excruciating pain in the bone. So sometimes they will have to do some biopsy to reduce the disease burden. So those patients, if you do biopsies and take those tissue, you're going to find that these tissues actually, they lose the glandular architecture. We know cancer has, in cancer you lose differentiation, but have a look here. Many of these cells are actually epithelial cells now, coming from bone, and they are expressing enormous amount of the androgen receptor, meaning as the disease progresses, the androgen receptor level goes up in cancer cells. So it is one of the major drivers of prostate cancer. And prostate cancer is not something which is restricted to only Western countries. There are lots of cases of prostate cancer in India these days. And what is believed now is that the cases are actually being diagnosed at much earlier. Previously, it was 70s and 80s and 90s. These days, you see patients coming to the clinic and having a positive diagnosis at even 45, 50 years of age. So this androgen receptor, which drives the disease, have a look what it does in order to drive cell growth. Basically, the androgens, which are sex hormones, the testosterone, they're located, they're circulating, they can get very easily accessed through the plasma membrane. As I said, they are hydrophobic in nature. The receptor, therefore, does not need to be a plasma membrane. The receptor in this case is cytosolic. What it is doing in cytosol, it is inactive. It is considered largely inactive in cytosol. But the binding of these androgens, you can see this is cholesterolic structure. These are steroid hormones. The binding of androgens can lead to protein changing in its shape. And it happens in biochemistry all the time. You have phosphorylation, it changes the protein from becoming inactive to active. In this case, this protein is becoming active as a result of binding to a steroid hormone. Once it activates, the receptor goes into the cell. It dimerizes and binds to specific parts on the genes, driving their expression. How many genes we are talking about? Thousands of genes. Their expression is regulated by androgen receptor. So it's very difficult, as Dr. Aziz was showing other day with those charts, network analysis. The genes are being expressed. What is their overall role? That is one of the challenges to establish. You have androgen receptor driving the expression of thousands of genes. What is the downstream pathways? What is the outcome in terms of cell growth? What is the outcome for cancer? What type of pathways do they regulate. And to make matter worse, what you're seeing on your screen, androgen receptor is amplified in prostate cancer, meaning there are more copies of androgen receptor. The gene is on X chromosome. So if you're a female, you will have two copies. If you're male, you have one copy. But sometimes you see even on X chromosome, there are up to 200, 300 copies, new copies of the androgen receptor created, which will eventually lead to more protein being formed, and as a result, the androgen receptor will be more active than it should be. And that's why on a IHC scan, it would look like that these cells actually have more androgen receptor compared to normal individuals. So I will stop here, and I am happy to take questions, Dr. Aziz.
Yes, thank you so much for setting up the post-lunch session, and we look forward to some questions from the audience.
I have to say that photo of yours, Dr. Aziz, looks like it's 10 years old. You're looking like a student.
Yeah, I got
Oh,
inspired
no, no,
from
that's
your
not.
photograph where I need some more growth.
More growth factor receptors.
Yes.
Yes, please feel free. If it is allowed, they can switch on their mics and
Yeah,
ask
we'll do
questions.
that. So
Khalida,
you
she's
can type if that is. I think the microphones are on, so they can already
okay.
do that. So in the morning session, we discussed all the techniques that are needed for multi-omics.
Okay, that's
The
good.
next generation
That's good.
sequencings and mass spec and the platforms, along with the data
That will
analysis.
make my job easier in the tutorial today then.
Yeah, so they were.
Thank you.
So they're at least familiar with the terms next generation sequencing, RNA-seq, and
Okay,
proteomics.
that's great.
That. So,
That's perfect. Then I can certainly try and complement that with the tutorial which I've prepared.
mhm.
So if there are no questions, shall we reconvene in, or
Yes,
more
we
than,
can.
we have 15, 14 minutes, so feel free to ask
Uh,
questions.
I think they're just 14 minutes left, so we can you can restart in 3. Uh, that's
Okay,
fine.
shall we stop then?
Yeah.
Okay, all right. Thank you. See
Anybody,
you.
if anybody wants to ask the question, they can just type in or pick up the
Yeah,
mic.
please type it. Feel free to ask as many
We are
questions
hanging
as you
around,
like.
but we are just giving you a break to absorb what
I'm
all
here as well.
Dr. Asim has just taught us. And I've already shared the YouTube links plus the folder link. So yeah, I've got all the material there. But I can tell you that there's no replacement for a one-to-one interaction.
I agree.
So don't think that everything is available online. So I'll read it later on. It'll never happen.
You,
Ask
you
your
don't
things.
get the same. That's right. Yeah, you don't get the same understanding of a subject.
All right, Dr. Asim. So we will be hanging around, but I'm muting now. So we will reconvene
Sure,
at
sure,
3 p.m.
sure. So Madiha is asking, can you please explain a little about the biochemistry behind the constitutively active receptor dimerization when valine changes to glutamine, as valine is hydrophobic amino acid and glutamine is hydrophilic. So I think I'm not a biochemist. I'm a cancer biologist, but I think what you're asking, your question has itself the answer. The changes in the so this valine is hydrophobic, and where would you find them? Normally inside the cores of proteins, while the glutamine is hydrophilic. And because of this, you might actually lose you can counter the extracellular domains, keeping it apart, perhaps. So they become really, really close in the absence of, if you lose those, first of all, the extracellular domains, or if you have these mutations, they might bring the monomers into close proximity of each other. And as a result, perhaps this can lead to initiation of the signal transduction cascade. I'm sure this must have been worked out, just that I'm not completely confident. How does it do so? Any other questions? Please feel free to type questions. I'll join you in 10 minutes again. I'm leaving my screen live. Shall we start, everyone? Dr. Aziz?
Yes, sir.
Yes.
Great. Okay. Thank you very much. All right. So that's where we've stopped. And there were some questions asked, some very interesting ones. Does androgen receptor always act as a oncogene or tumor suppressor as well, on similar lines? So as you can see here, that androgen receptor is a driver of prostate cancer. Several years ago, and current research in my lab is trying to understand whether there are any roles of androgen receptor as being a tumor suppressor, but it largely looks like it acts as a tumor promoter by driving a gene expression programs. Which genes are being expressed by androgen receptor? You can study RNA sequencing to establish those gene sets which are driven by androgen receptor. Uh, no audio, Yusra said. Are you able to not hear? Can you hear me?
So for me, it's working fine. Can anyone
Okay.
else confirm it?
Okay. Thank you, Padmavati. Great. So we are trying to understand how these pathways are regulated. And in fact, the function of androgen receptor to serve as an oncogene versus a tumor suppressor could also be driven by whether we're talking about cancer or non-normal cells, but also the positioning of the androgen receptor. Um, this is highly confidential part of a patent, so I'm not going to talk about it, but I can offline touch base with you if you will be interested. So because this is androgen receptor is driving this pathway, and in breast epithelial cells, what you also see is that the similar androgen receptor can drive specific forms of breast cancer, such as molecular apocrine type of breast cancer, but also estrogen receptor. So estrogen receptor will actually drive a similar signal transduction cascade in breast cancer epithelial cells to drive their growth and proliferation uncontrollably. And therefore, both androgen receptor and estrogen receptor are drug targets. They are the therapeutic targets in the clinic by drugs like enzalutamide, which can block the androgen receptor activation, and drugs like tamoxifen, which can block the estrogen receptor activation. And most of the time, these drugs are actually quite effective at curbing the tumor growth. As you can see from this PET-CT scan, there is an individual with prostate cancer, and you can see the cancer growing in, it is invading locally, as well as you can see some cancer in the bone. But you can see 2 weeks after the therapy, the cancer is looking like there is hardly any response. But have a look what happens when this person is on androgen receptor targeting therapy. The cancer, these shadows of cancer, they're largely gone. So these drugs are effective. The problem is they're effective on a time scale of uh, good few months to perhaps at best a year. Eventually they fail. So the challenge is for the next generation, for you to understand why this happens, and develop better drugs that can actually sustain in a sustained fashion block the androgen receptor activity. So this is one of the things I wanted to talk to you about. The other would be the fusion of chromosomal arms in order to give rise to hybrid genes. For example, this is what happens in a CML, which is a form of blood cancer. What you see if you take slides from these patients, immunocytochemistry slides in pathology labs, what is prominent is this presence of a new chromosome, which is otherwise not found in a regular people. This is called Philadelphia chromosome. And as a result of formation of this hybrid chromosomes, two genes come together, BCR and ABL. And that actually can drive the abnormal growth of lymphoid cells in a CML disease. BCR-ABL is going to be constitutively active, meaning it has lots of switch on and off alternator button. The BCR-ABL kinase is always active, sending the signal transduction to become always active. The cells are actively proliferating into this cell cycle, and as a result of this, manifest themselves into this form of cancer. These sort of diseases, I think, are probably much better. We are much better to treat them. Some of them are, in fact, treated, not like prostate cancer, where you have 5 months, 6 months, perhaps a year on a drug, and then the cancer comes back. In this case, sometimes the cure is really good. So the person dies of a different disease than the cancer itself. And because these genes are not present in normal cells, for example, these fusion genes, sometimes you don't really see a lot of side effects of treatments like this. Uh, so Padmavati is asking questions at 2 weeks of the therapy, the disease looks to be aggravated. You have a point certainly, what could be the reason? The reason could be that there may be a feedback switch on, or this is just an individual specific, that it takes time for cancer to start showing effect on so the molecular changes may still be happening at 2 weeks, but they are not really translated into the growth of tumors. And by the time we go into the therapy, there was no scan for 4 weeks. So perhaps you can see that if this was there, that after 4 weeks, the tumor started to disappear. And that's missing basically from this. But have a look at this paper. The reference is provided. But very good point, Padmavati. So this, as I mentioned, if you have these tumors being driven by these proteins, which are fusion proteins, the treatment sometimes can be much better. And it does not have so much collateral damage in terms of side effects, because normal cells actually will not have these hybrid genes. So this has been used, and have a look at this drug called imatinib, which actually it does exactly the same thing. It binds to this ABL kinase, and by doing so, it can actually block this kinase ABL, BCR ABL, to function. And you can see that drug itself is super efficient. You can see before Gleevec, you see a big gastrointestinal tumor. And 1 month after the therapy, of course, if the tumor was driven by this kinase, you can see that the cancer is largely gone, and all you see is normal bladder. But the problem we face is the ability to transfer uh, this finding. This is not a representative efficacy slide, if you understand what I mean. This happens in a lot of patients. For some diseases, it's very rare that you will see extended efficacy of this sort. But for many orphan diseases where there is no treatment, basically nothing works. And please don't forget, many of these tumors are heterogeneous, so they have a tumor heterogeneity. Within the same tumor, some cells are being driven, the growth is being driven by some drivers, for example, driver A. Within the same tumor, there is heterogeneity, so within some cells are being driven by driver B, and there may be a third subpopulation which is being driven by driver C. So if you take a tumor sample and you don't take a representative tumor sample, you take part of it, miss part of it, you're only going to see, okay, it looks like only driver A is present. And based on this, the medical oncologist may start therapy targeting driver A. Well and good. After subsequent treatment biopsies, if they are taken again, it might look like, okay, the cancer is being driven by driver B and C. Well, this was the case from the beginning. What you have done by blocking driver A, you have allowed those cells which were being driven by driver A to disappear or decrease in number, and now the tumor is predominantly driven by even more nastier drivers, driver B and C perhaps. And that's why if you do sampling now, it might look like that actually there are now new drivers of the same disease. While we know most of the human cancers, adult cancers, are in fact driven, they have an endogenous inherent tumor heterogeneity. And that is a feature of cancer that makes it very difficult for medical oncologists, for the doctors, to treat cancer. So this was about the drivers, the oncogenes, some of the oncogenes, and they come in all shapes and forms. I discussed some of them with you. What about viruses? They can also cause cancer. And this is such an important discovery that the Nobel Prize was awarded on the discovery of the sarcoma virus of connective tissue, chicken sarcoma virus that causes cancer in chickens. And there was a very simple experiment done. Chickens would develop this sarcoma in the breast muscles, and it looks like this was infectious. So if you have this in populations, others will get it as well. It turned out that, so what Rouse did in the experiment done at Rockefeller in New York is take out the tissue, cancer tissue, and ground it up to make sure that you can actually get, if there are any agents which are causing cancer. Previously, it was not known that cancer are actually transmittable. It was thought that there may be genes underlying that is causing cancer. But it looks like what they did with in this experiment, use this, make a very fine paste of these cancer cells and filtered them, and getting a very clear supernatant here, nothing here, and injected them back into chicks, and identified as they grow that actually they formed also cancer. And that was one of the first very important reports showing that cancers can be transmittable. And what is driving in this case the cancer is again an oncogene. It's called SRC. I've seen in the textbook there are V and CSRC. We have counterparts in human cells, which is called CSRC, cellular SRC. But viruses also carry genes like SRC, which is a viral form. So you will have initials VCSRC in those cases. This is not virology lecture, so I'll keep it brief. But you can see here what happens when the virus goes in cells. This is an electron micrograph. They hijack the machinery of a host cell, and in doing so, sometimes they start producing their own copies in great numbers, which is a typical example of a lytic cycle. And you can see lots of new viruses are putting out of the infected cells. So this would be a classical example. Many viruses, how they cause such cancer, they carry lots of times these oncogenes, but sometimes they can actually stop the good genes in our body, tumor suppressors, which I'm now going to start. They can stop the good tumor suppressor genes from functioning, from saying there is something wrong with cells. So you can see there are 3 viruses shown on this slide. There are actually many more. You can see here, this is SV40, and they have a specific proteins, in this case, large T antigen. Adenoviruses, proteins like, they don't cause cancer as such in humans, but flu is attributed to them. And the famous HPV, and you can see they contain different proteins, E7, E6, large T antigen. Many of them are actually involved in blocking the function of good genes, which keep us safe, which we will start in a minute now. Uh, so viruses are, you can see, can give rise to transmittable tumors. The alarming thing is that we only know about a handful of such cases. There are reports that viruses particles have been found, viral DNA has been found by some in even prostate and breast cancers. Those studies need to be validated, but there is very limited extended data on such tumors. So it looks like that there may be, we are sitting on the tip of an iceberg. There may be other transmittable cancers attributed to viruses as well. So the other feature of the cancer would be that the good genes, they become active. We call them tumor suppressor genes. The cancer cells don't listen to good genes. In other words, they can evade growth suppression. So this is very important. The good genes, what I'm calling the tumor suppressors, are actually much more higher in function. They have an upper hand in deciding whether a cell is going to become cancer or not. And I'll tell you in a minute why this happens. So have a look here. These are the genes which are there in cells, but they don't get active all the time. Are they only active in tumors? I'll show you that is not the case, but they are there to make sure that any cell which has an oncogene, previously they were proto-oncogenes, if there are mutations and the cell takes a first step, perhaps by enhancing the rate in which they enter the cell cycle, these tumor suppressor genes can sense that activation of oncogenes. And they try to repair these defects in oncogenes, converting them back into proto-oncogenes, getting rid of them, the process called apoptosis. So as long as these are there, the tumor suppressor genes, we don't have to worry about developing cancer. And have a look why they are there. Are they only there in cancer? You can see in this embryo, there are genes, tumor suppressor genes, which are very important. So embryos do express genes like retinoblastoma, APC, but most importantly, the function of P53 is very well established in cancer. And what happens in apart from cancer is that they carry out a very important role in the development of embryo. Why? Have a look at this slide. What you're seeing as an embryonal hand, the development, and you can see here in this slide that there are these black dots. Can you see them? Basically, what it is telling you are the staining for the P53. So these are the points where P53 is becoming active. And why this is needed? If p53 will not become active, we will not get our hands. We will not be able to write anything, document history, and make uh, arts and so on. So you can see, even for the development of embryo, the tumor suppressor genes are so important, because their major function is in carrying out, in this context, is to carry out apoptosis and remove these barriers between the fingers that lead to us having these nice beautiful fingers. Without them, that would not have been possible, and we would have ended up having webs. So what happens in cancer is that these genes sadly are mutated as well. And when they get mutated, this time it will not be a gain of function like happens in oncogene. This will be a loss of function. So sadly, they will not be able to function properly, and that can cause cancer. But you can see equally, they're very important for the proper development of the embryo. So there are some questions. I'll take them in the last 15 minutes. Please feel free to, well, Dr. Aziz has also answered, but yeah, please feel free to leave them until the last 15 minutes, and then I can take those which remain unanswered. So you can see here how important these genes are, and how do they become active? You can see that they can cause massive apoptosis. They can cause massive cell death in embryonal stages. Of course, this is needed for metamorphosis, for the development of, in this case, proper hands, and similarly other organs as well. So how do these genes become active? There are lots of things which can activate p53. You may see in slides, and this is one of the exceptions, that p53 is the name of the protein, but the gene for p53 is called TP53. Usually you have the same name for gene and protein, but in this case, the gene that codes for p53 protein is called TP53. So have a look what happens. Any signal, in a sense, that leads to the damage, that poses a threat to the DNA, can actually activate p53. And when it becomes activated, I was giving you example of epidermal growth factor receptor. What happens is that needed in order to activate the receptor. Same was the example of androgen receptor. You need the dimer to bind to the DNA in order to drive transcription. In this case, it's not a dimer, it's a tetramer. So you need four copies, essentially identical copies of p53 protein to come together to form this active protein. And therefore, you need a lot of different, you can see here different signals are needed. And you can see p53 can be activated by lack of nucleotides. What will happen if you don't have nucleotides? Well, they are the building blocks of DNA, aren't they? So if you don't have them, it is going to cause defects in the DNA. UV radiation is a well-known damage to the DNA. Ionizing radiation, likewise. Oncogene signaling, that is important for us. So what I was showing you in the previous session, if you have any oncogene signaling, p53 will become active in cancer cells. Hypoxia is also known to activate. So less concentration of oxygen can also activate p53, and the block of transcription. All of these factors are some of these examples which can allow p53 to become stabilized, make a homotetramer, so four copies of the protein come together. Now, p53 is very smart. It does not lead to apoptosis straight away. It does not kill the cells straight away, as you saw in this example, that there was an apoptosis taking place here. In fact, p53 is one of the good genes because it tries to manage the damage. How? First of all, it tries and blocks the cell cycle progression of a cell. So that allows cells, what P53 does by doing so is allow cells to repair the DNA damage. So I was showing you some examples yesterday of base excision repair, mismatch excision repair, nucleotide excision repair, and I showed you there were lots of different proteins, genome maintenance protein, we call them. Many of them, their expression is under control of P53. And you can see here why this is important, because by blocking cell cycle, the P53 wants to give time for a cell to repair any damage to DNA. So P53 is, of course, trying to mitigate this damage to the DNA. Not having it can cause cancer, but if a patient is being treated through cancer therapy, for example, radiation therapy, do you want P53 to become reactive? No. So in cases where someone has full-blown cancer, they're being treated by a therapy which will intended to damage the DNA, chemotherapy, platinum drugs, radiotherapy. In those circumstances, it is noted that tumor suppressors can become reactivated, causing therapy to fail. It doesn't happen in all patients, but there are different reasons because of which therapies can fail. One of the examples will be this one. There is something else that P53 does, is that it blocks the progression through the angiogenesis, formation of new blood vessels, because really what cancer is trying to do is, by the pathway I was showing you yesterday through which the cancer migrates from the primary site to other sites, is make new blood vessels that allows cancer cells to traverse far away into the body through the blood. So one of the things P53 also does is to block the angiogenesis. And if nothing works, it does not really help the DNA. The damage to the DNA is beyond repair, which is what the radiotherapists intend to do in the hospitals, is to damage the DNA of cancer cells beyond repair. Then the P53 will cause those cells to die. So all these signals activate P53, and P53, you can see this is very important, so important that this is called guardian of the genome. So what happens as a result of when cell cycle is arrested? Some cells might become senescent. They just don't divide. Is it good? Is it bad? Sometimes it may be bad, because some cells, they just stop dividing. They think we are being killed because we are dividing. Let's not divide. You know, the therapies, many of them work because cancer cells are dividing very, very fast. They just stop dividing. And once the defect is corrected, they will return to proliferation. And as I said, if the damage is beyond repair, the cells will undergo apoptosis. So when I said that ionizing radiation, any threat to DNA can activate P53, I'll give you a classical example. What happens here? Have a look here in this diagram. P53 becomes phosphorylated and stabilized. In its unphosphorylated form, P53 is not stabilized. It is degraded by MDM2 pathway. You all know this. This is classical textbook molecular biology. What activates P53 is DNA damage, one of the things. And this is DNA damage which is sensed by this protein called ATM. So ATM is a kinase. One of the things ATM does, it senses any DNA double strand breaks and sends a signal to P53 in the form of phosphorylating it and stabilizing it to allow it to carry out the role I showed here, one of these rows here. Now you might want to ask, how many substrates are there for ATM? Is it P53 the only substrate? Well, we know about 800 proteins are phosphorylated by ATM once it becomes active. So you can see how enormous the role of ATM is. I'm only touching on one very brief pathway here, which is ATM-mediated activation of P53. And once activated, you can see here what P53 is trying to do. It is blocking the cell cycle by making proteins like P21. If this is good enough, by blocking cell cycles, it allows cells to bind precious time that will allow the DNA to become repaired, well and good. But if that doesn't happen, the last resort P53 has is for the greater good of the whole organism, and the cancer does not manifest. They will allow those cells to actually die. Now, we call P53 as being the guardian of the genome. Have a look at this slide. You can see as many as 50% of the women who have this ovarian cancer, but also our population with esophageal cancer, colorectal cancer that Dr. Aziz studies, have a look, 45% of the patients will have TP53 mutations. This is enormous, and that tells you something very important. As long as P53 and other tumor suppressors in other cancers, it is all context dependent. I'm giving you one simple, simplified, should I say, example. As long as they are intact, as long as they are not mutated, it's very hard to get cancer, despite having oncogenes. Because even if there are oncogenes, cells can sense those presence of oncogenes and will allow these tumor suppressors to become active, and in doing so it can actually kill a cell in which there is any which is going to cause any future threat in the form of becoming cancer. Now compare this with an oncogene. For example, lung cancer. Lung cancers is driven by different mutations in different oncogenic drivers. So these are some of the oncogenes. Many of them are kinases. You can see here 25% are driven by KRAS, 17% EGFR, ALK 3%, 5% MET, 3% HER2, 2%, and so on. The numbers are too little. So if you start to stratify, to divide patients based on the expression of these oncogenes, it's going to be very difficult to treat a patient actually, because many of them might have KRAS, but in addition they may have EGFR as well, they might have HER2. It's very difficult. So the cell is using different oncogenes, in this case you can see, to drive cancer growth, a cancer cell. But you can see here as many as 50% women who have ovarian cancer would rely on TP53 to not develop cancer. So if you can see, there is a big proportion of women which are kept safe by from developing ovarian cancer because they have good TP53, and that's why the mutations which cause TP53 do not function are so common. So it does not really matter what sort of mutations you have in oncogenes or proto-oncogenes causing them to become oncogenes. As long as these tumor suppressors are intact, normally a cell is very good at taking care of these threats which come in the form of mutations in the DNA. So I hope that is clear. We still have some time. I will start taking questions after this session, so please bear with me. Going to the next slide, I'm going to tell you what sort of mutations are found in TP53 in bladder cancer. Have a look here. There are all sorts of these mutations. These are mutations in exon. So this is the P53 gene. You can see here, these are the mutations in exons. These are point mutations shown here. These are silent mutations, and these are splice site mutations. But you can see here there are three parts of this protein as a transcription factor. I was giving you example of androgen receptor. Remember, androgen receptor is a transcription factor. So is P53. Why? Because it binds to DNA to drive mRNA expression of specific genes. It drives transcription. What should they have? All transcription factor, a domain which is very common is the DNA binding. So they need to bind to DNA in order to drive transcription. So you can see all these mutations which are found in cancer, basically majority of them affect the ability of P53 to bind to DNA. If it cannot bind to DNA, it cannot make those proteins which are needed in order to block cell cycle progression, which are needed in order for the DNA to be repaired, which are needed for the cell to carry out. apoptosis. So you can see here how important the p53 is. There are many other oncogenes, but the p53 stands out because of its extensive role in different tumor types. So there are groups like this one who tried to restore the function of p53. Function is lost, p53 function. So they tried to restore to reactivate p53 by using this compound CP53 in skin cancers, and to some extent they were successful, at least in experimental conditions, showing that the treatment administration of this compound to mice actually can reactivate p53 dependent function and save those mice from getting a worse disease, skin cancer type. So in fact you can do that, but you can only do that if you can ask yourself, if you Google this, how successful has this been? You can only do that if there are mutations which you can mitigate by this drug. Sometimes you might find there is a loss of expression of p53 due to the genomic loss. So the gene altogether is mutated, is gone. Basically, what you can restore them? Nothing. But most of the time, you have these mutations, and if you have compounds like this, which can actually restore the function of protein, which is good news, in order to make treatment work better. Which kind of treatment? Not radiotherapy, because radiotherapists, they're trying to overburden cells with an enormous amount of DNA damage beyond repair. And if you activate p53 in them, it might allow cells to actually get DNA repair pathway activated. You don't want that. So this is certainly very important. There is a question I can see, and Sandeepan, I would have a slide. Sandeepan is asking, it was seen elephants have more copies of T53, making them immune to cancer. If by any means we try to mimic the same in cancer by birth, it will be viable in the first place. Sandeepan, I'm coming to your question. Have a look at this slide, Sandeepan and everyone. It looks like P53 keeps animals safe, such as elephants, these giants, from developing cancer. Um, why? And do they live any shorter than humans? Of course, we get cancer because we live long enough. We have lots of cell division. We saw that yesterday. The cells are dividing much more faster. In some tumors, they have more propensity of developing cancer. Some cells don't divide at such a fast rate. The cancers are rare, rather. In this case, animals actually live as long as average human. Despite that, there are no cases reported in humans. You can see here 25% of humans will develop cancer, while elephants, they don't develop any cancer. It turns out they have many copies, as many as 19 to 20 more copies of p53. And as a result of this, they are kept safe from developing the cancers. They just don't develop it. You might ask, okay, how do you put an elephant under a scanner, an MRI and a scan to see whether they're developing cancer? Well, not every human being would go to MRI scan to prevent them from cancer. You go to a doctor when you have any problems. Elephants don't get those sort of problems that will be obvious indications they are actually developing cancer. Some of them can be detected by blood-based assays as well. So in fact, they don't develop cancer. And what happens here is, Sandeepan was asking this question that can we try this approach in human? Well, it might be that if there are some humans who, as an anomaly, defects in the genome, may have more copies, may actually survive longer, may be immune to cancer, but that's usually not the case. You don't have very many copies of p53 in humans. So why elephant? Nature gave elephant. I thought about this question a lot. Why nature gave elephants so many copies of p53, not to us? And I think, and this is my personal view, I think the answer is that elephants are really, really huge organisms. They can afford to have a lot of cells undergoing apoptosis. That is one of the primary functions of p53 in embryonal stages in normal non-cancer cells. They can carry out apoptosis. If you have 20 times more apoptosis in an elephant, what is happening? They may still withstand this extensive loss of cells. Humans, as opposed to elephants, are not so big. They may not be able to be saved from this so much loss of cells. They might actually become um, uh, different type of illnesses, have developmental disorders if nature gave us so many copies of p53. So I would say that one of the arguments is for our own good, so that we don't have atrophy of different organs. The nature did not give us so many copies as it did to elephants. And of course, there are other differences like diet and so on. They don't eat junk food, do they? It's like us. So, and many other things, of course. So I hope that will answer the question.
Can I add here something?
Yes, please. Yes,
Rasan?
please. Yes,
Uh,
please.
this is a very nice observation, and there's another one that happens in Wales. The Wales, they have got only one copy of p53, and they live for hundreds of years, probably more than 100 years, and still they don't develop cancer. There are no known cases of cancer in Wales. So it was found that p53, very good. It's a guardian of the cells, and it's holding back the tumor suppressive gene. But whales have got a very good and rapid mechanism of DNA repair, because there the cells are constantly dividing, and they have to keep up with their energy as well. So they have got a lot of faster processes that are going on. Probability of having cancer with in the whales was much higher than in elephants, and they have got only one copy of p53. Still, they don't develop cancer. So there are alternate mechanisms that are keeping them safe. That's what I would like to add.
That's a very interesting and excellent point you've added. I think if they don't have very much p53 and they have very much robust system for repair of DNA, I would think, am I correct that they don't develop as much variations as humans do? Because part of the DNA that we develop mutation is to make us more adaptable and evolutionary successful. Is it possible? Is it the case with whales that they have a lot of adaptation abilities, or they have a very small population which is very genomically synchronized, that they don't adapt, they don't evolve, perhaps? So there is a trade-off, I think. If you have a system which allows you to repair DNA very, very quickly, you may not actually evolve at all. Like some fishes I know, they remain alive for 300, 400, 500 years, and there are no changes. They don't evolve at all as a result. So perhaps they have a very excellent, I would say, DNA repair system, but then it comes at a cost of them not being evolved as humans have. Who knows?
Yeah, this again reinforces the concept that every species is unique. Every species has got their own mechanism.
Right.
Let's deal it that way. So interesting discussion. I think anybody else would like to jump in and add to it. Thank you.
Okay, so I will, if there aren't any, then I can carry on with more questions. We will start taking some questions. Padmavati, I see there is a question. Let me quickly finish this if I can. Have a look at this paper. I was showing you this paper yesterday, and it came back again. Antioxidants accelerating, they're promoting antioxidants, which we buy in the supermarkets, especially in these countries in the West. Antioxidants, they're good for you. That's what we have been made to believe and deceive. Basically, this is one of the excellent examples, this publication showing that antioxidants used in this paper actually can degrade p53. So instead of stopping cancer, they can actually accelerate cancer. So my personal view is I don't take any antioxidant supplements. I get it from my food, my fruits, vegetables, and so on. So that would be one example of it. So the other disease. Somebody was asking yesterday, Dr. Aziz actually himself was asking about retinoblastoma. You can see here this disease. And I should tell you something. There are cancers. Sometimes I think there are lots of cancers which we can treat with where the treatment is available. We know there is a standard treatment, but because of the healthcare system, the infrastructure, those treatments are not available to human beings at par with every other nation. So have a look here. This individual, this child, which was identified to have this disease, retinoblastoma, at much earlier in Canada, and was successfully treated before, sadly, a child in Africa where the tumor actually became full bone, the retinoblastoma invaded into the brain. Which is a bad news, of course. So you can see here from the initial diagnosis, very early stages to very late stages, the cancer can actually progress. But the treatment is very successful here because it's a childhood cancer, which is mostly germline related, meaning they inherited bad genes from their parents. They were not as a result of some mutations in the children. They will not have the possibility of those sort of mutations happening in such a short lifespan that they have lived, but they actually inherit, sadly, germline mutations, bad copies of the uh, proteins, genes from the parents. So basically, this is how it is formed in the back of tumor here, back of the retina. I'm sorry, and you can see here this can actually manifest itself to such horrendous conditions when it is fully grown. And you can see here there's another transcription factor that regulates this, and there is another tumor suppressor that regulates this formation of this tumor. We call it retinoblastoma. This is mostly in, you can see in childhood cancer, this is very common. And you can see here what is happening. P53, we saw was becoming active as a result of phosphorylation by ATM in the charts I showed you previously. So phosphorylation was making P53 active. It was able to form tetramer, bind to the DNA. In this case, what is happening is this tumor suppressor is being regulated differently by cyclin CDK. Now, these complexes are very important. Cyclins are proteins which regulate the entire cell cycle of cells, as you know. So what happens is the cells decide to divide. They have transcription factors that allow them to carry out this. And one of the most important one is called E2F. So E2F is a transcription factor. If I ask you what is the most common domain you will find in a transcription factor, is a DNA binding domain, right? So it cannot bind to DNA every time. Why? Because it is being trapped, it is being stopped, it is being inhibited by this protein called retinoblastoma. It binds it tightly, and by doing so, it stops the E2F from becoming active. Dr. Aziz, shall we take a break and then I can conclude this session, or do you want me to carry on? I have a couple of more slides and that's it.
Well, yes.
And then I can take questions. I just don't want the students to become overburdened. Done 45 minutes. So let me finish this anyway, if there are no questions. Is that okay?
Yeah, because this will be followed by a tutorial, right? So
that will be then the tutorial. I have one more slide and then we can start the tutorial.
Let me give a break and then.
Okay, we can finish this session and then
it's going to be for 2 hours. So you can take your coffee.
Yeah, I understand. Yeah. So shall we meet at?
Yeah.
So let me finish this after this session, and then in the meantime, I can answer the questions which were being asked. Perfect. Okay, so let me have a look.
So shall I send you some coffee?
No, thank you very much. I don't drink coffee.
Okay. So you have Cafe Nero and Starbucks both, right?
I don't think when was the last time in the last decade I had to go to. If I'm on an airport, I might have some tea, but that's not really the tea. You need to boil the tea in the hot water to make the real chai. So,
Yeah.
yes, so there are questions I can now attend to. Padmavati is asking, we observed such increase in cell proliferation at a lower concentration due to phytochemical screening. And okay, that's true. If you use a suboptimal doses of uh, some drugs, actually you can see an increase. That's true. So that's why it is very important to titrate and establish a maximum tolerated dose that you can give to a patient without causing much severe side effects. So certainly that is a good observation, Padmavati. Khalida is asking, cancer caused by, okay, and this has been answered already. Uh, Sandeepan is asking, I believe Sandeepan, I've answered your question, and Dr. Aziz as well. Saida is asking, okay, let's see, admin. Padmavati is asking, while it's naturally present in elephants, why is it mentioned as a transgene, sir? What does it signify? Very good question. On top of the head, I don't remember. I think this, because this is not really a CP53, meaning a cellular, although it is present in the cell, it is in terms of its regulation, there may be that it is constitutively active. I know that this P53, which we call transgene, actually does carry out DNA repair. So the function is very analogous. Why the different name? I'll have to go and check. But from what it looks like, from what I know, is that it is able to carry out analogous functions as does the cellular P53 by safeguarding the genome. It's just a different name, perhaps because it is a product of transposons, making more copies, amplifying the genomic locus where this P53 is present. Perhaps that is what is signified by the name transgene. So I think that's pretty much it. Shall we join in 10 minutes?
Yeah, sure.
Okay. I'll see you soon then, everyone. Please feel free to keep on posting your questions so that we can discuss them before we jump onto the omics.
Would there be any need for tutorials? Oh, sorry.
Sorry, go on.
Do they need a different
sure.
system or any other apparatus for the tutorial?
I think so. I have some slides
Okay.
that I can first go to, and then I can show them some platforms, because it is being recorded in their own time. They can go through some of these platforms and gain the experience that they need. Somebody was asking, I think Khalida was asking about computational biology. Of course, I think it is an excellent career. What I do suggest people who want to do computational biology as a career, they should be really, really smart in mathematics. Without it, they will not be able to efficiently code. And some people are really super smart in mathematics and actually do develop to become a good computational biologist. We in fact need much more computational biologists now that we have a lot of data, astronomic data coming from different labs. We can sequence, yes, but how do you make sense of this large genome sequence data from 3,000 patients? You need good computational biologists to analyze, make sense of that data, and those are the individual scientists which will be in much higher demand than anyone else. So one needs to think about that. But yes, please do see where your passion lies, because what we do, Dr. Aziz, myself, you can still, as you can see, become an academic if you have a passion for wet lab science, because there are lots of wet lab scientists these days. But computational biology is a very fast developing area. You need to have the right skill sets and inherent interest in mathematics, coding, in order to become one. So Arham is asking a question. A new drug is made which takes approximately.
I can take that if you allow me, and
yes,
you can
please
take
do.
Correct, yeah.
Yeah. Okay, thank you.
So Arham, it's not about inventing or discovering a new drug for every person. It's about matchmaking. The drugs and molecules and biologics, they are already available, and there are millions of them. what we have to do is to find the right drug for the right person at the right time. That's what we mean by personalized medicine. Don't get confused. This is a very common confusion that we will be knowing the target and then we'll be developing 10 to 15 years, or even if it is accelerated for a couple of years to develop a new medicine. No, the molecules are already there. We need to give it to the right person. Okay. If that answers your question, otherwise you can further ask and Dr. Asim can enrich that as well.
Any other questions?
No, I don't see that. It's still time, 5 minutes. I would encourage the participants who are at that stage, they can also ask Dr. Asim about their career-related questions, like somebody did about the career in computational biology. But if you have other questions regarding the internships or PhD positions or postdocs or scientist positions, I think Dr. Asim would be able to give you an overview or some some guidelines, some pointers that will be helpful
Yes,
for your
please
career.
do. Yeah,
Yeah.
I'll be
So,
delighted
from
to.
yeah, apart from the science, utilize this question answer session for other things as well. Okay, thank you.
Okay, that's great. So, shall we resume and finish this before we switch to the omics tutorial for today? And as Dr. Z said, please feel free to ask me. Open your mic. If you want to show yourself, go ahead and discuss if you have any career-related queries. Um, okay, so what you're seeing here, what is happening in this disease, the cancer of retina, is basically one single gene mutated sometimes, and what it is the loss of function mutations in retinoblastoma, which I said is a tumor suppressor. So normally this pathway is regulated highly by and coordinated by the cyclins. So cyclins needs to become activated by binding of the cyclin CDK complexes. And one of the activation signal is the loss of P50, P16 protein. And when this stops binding to this trimer, the dimer becomes active. So you can see how much variety is there. Sometimes proteins might become active as a monomer. Sometimes they need to be tetramer. Sometimes they need to be dimer. Sometimes they need to be phosphorylated. Sometimes they need to be dephosphorylated in order to become active. And all it lies is in the changes in the conformation of protein that results from such complex processes. And see what is happening here. In the when the cells decides to divide, and before that, of course, the DNA needs to be replicated. So what happens in a G1 phase of cell cycle is that complex becomes active, and the activation simply means that now this kinase can phosphorylate a retinoblastoma, but it is only happening in G1 phase of cell cycle. And this phosphorylation actually makes a retinoblastoma inactive, this phosphorylated retinoblastoma. And that allows E2F to become active. When E2F as a transcription factor becomes active, uh, what happens is there is expression of genes. Which genes? Genes that are important for DNA replication. In fact, DNA polymerase, we discussed this previously, the proofreading function, if you remember. DNA polymerase, the expression is under control of E2F. So for DNA, for a cell to prepare for a division, it needs to have the entire DNA replicated. And thus, this replication requires these proteins, cyclins. And these proteins, how do they do that? Is by allowing E2F to become active. How does it become active? When retinoblastoma is phosphorylated. So remember, it's the same name of the protein or the gene and same name of the disease, retinoblastoma, as I showed you on the previous slide. So what is happening here is retinoblastoma needs to be phosphorylated by this complex. And when this complex becomes active, retinoblastoma is phosphorylated. E2F can now drive the transcription of genes, which we need for DNA synthesis. And it has been found that there are germline mutations, meaning mutations that passed on from parents into the little poor child to have mutations in retinoblastoma. What sort of mutations? The loss of function mutations. And as a result of these loss of function mutations, retinoblastoma cannot efficiently capture E2F when it should not be active. And as a result, E2F is always on in both conditions. And remember the switch I was showing you. And once this is on, you can imagine this can lead to diseases like we discussed here, diseases like retinoblastoma. In Western countries, hardly anyone dies of retinoblastoma. Do you know one of the reasons? It is early detection. You can detect the cancer much earlier than it reaches to this stage. But also, retinoblastoma does not have a lot of mutations. Usually, the mutations which you find in a retinoblastoma are restricted to the RB gene itself. And these mutations are only handful. They're not very many. And there aren't other oncogenes, there aren't other genes which are mutated, only this one. And this is why the mutational noise of this cancer, meaning how many background mutations are there in a 3 billion genome, they're very little. They're normally restricted to one gene. So the mutational overall noise is much more restricted. You will not send a patient with retinoblastoma for whole genome sequencing because you know that most of these mutations are restricted to just one single gene. And therefore, because their mutational profile is super restricted, the mutational noise is very, very little, these tumors tend not to metastasize because metastasis, as we discussed yesterday, requires a whole new set of different mutation happening at different times of a life cycle of a cell and its products that will take a very long time. And that's why cancer diagnosis is usually very common as we grow older compared to children. So you can see there is an element of mutational burden. If there is a high mutational burden, like in many diseases, prostate cancer, breast cancer, compared to this one where mutational burden is super, super low, you can see that the treatment of such cancers will be much more straightforward. The prognosis, the outcome of the therapy looks much more positive if the mutational burden is very, very low. Compared to those cancers where there is an extensive changes to the genome, how do we identify those changes? One of the things is to do RNA sequencing that Dr. Aziz discussed earlier in the day today. So that is one aspect I wanted to touch. So to summarize this, you can see here there is a cooperation going on in normal cells. There is a regulation where proto-oncogenes are promoting growth. They can reverse injuries. In terms of having injuries, of course, proto-oncogene functions become heightened compared to the normal levels. And tumor suppressor genes are blocking any unregulated growth. But what happens in a cancer cell as a result of mutations, which mutations, gain of function mutations in proto-oncogenes, which transform them into oncogenes, and loss of function mutations into tumor suppressor genes, which allow them to lose their function. If you combine the effect of this, the cell will have an overall abnormal growth. And that is the first step on a cancer pathway. So they will have a lot of barriers to cross. We discussed a lot of these barriers yesterday in the sessions we had, and that's why those tumors which actually metastasize to distant organs, they will need to have a lot of mutation burden. They will have a lot of mutations in very different pathways to carry out this journey from the primary tumor, dissolving the base membrane where the epithelial cells are sitting, moving into the connective tissue, gaining access to the bloodstream, surviving in the bloodstream, surviving the immune system, then going and getting out of the blood vessels, trying to find the right organs, traversing through those organs and making their colonies. So they need to be super fit to carry out these journeys, but that comes at a cost to the host organisms, and that comes as a result of a number of mutations which make them fit. And that's why you will have a much heightened mutation load in tumors which are metastatic compared to the tumors which are still at primary site. So in a disease, it is possible a liver cancer or a colorectal cancer, if you open up a patient and have a look at the primary tumor in large intestine colon, you might find the mutation profile is rather restricted compared to the same colon cancer which has gone into liver. They may have, in addition to the primary mutations in the primary cancer, they may have a new signature of mutations that allowed them to invade and metastasize into the new organ and sustain their colonies there. So mutation overload, how much mutational burden is very important in a cell's life on a cancer pathway. The more is the mutation load, the chances are the cancer is going to get nastier. And it may not be sufficient for a single drug to function to restrict its growth. So I hope that is clear. Here is the summary for this lecture. And before I move on, I would want to go to the questions. If there are any questions that you want to ask uh, on what we discuss in terms of the uh, tumor suppressor genes or the other gene, the oncogenes. If there are any questions, please feel free to ask me now. Otherwise, we can simply switch to a discussion on this tutorial on omics, which I'll try to keep short, of course, because a lot of it has been very elegantly discussed by my colleague, Dr. Aziz. Well, if there aren't any questions, please feel free to. Okay, there is a question now. Padmavati is asking, and please feel free to ask me any career-related questions. She finished her PhD in cancer research in IIT. I found your research. Thank you very much for your kind words, Padmavati. Uh, uh, Postdoctoral opportunities. So at the moment, I don't have any availability for a postdoc in my lab due to the funding crunch. So I don't have the postdoc I had in the lab. Recruiting a postdoc is actually quite difficult and different in this country because they need to bring budget to cover the indirect cost, meaning the electricity, the heating. It's a cold country. In India, most labs don't really consider the government. In fact, does not give any indirect cost. But in countries like UK, US, you need to have substantial indirect costs. So at the moment, I don't have it, but I can tell you something. If you go to this website, you have the right profile. You will get a response positive, I hope. But also, if it is not successful, for any job in UK, you will get an answer whether your application was successful or not, which is quite good because once you apply, there are many countries I have heard where people don't even respond to emails. So at least if you're applying in UK, I can tell you, you will get a response after the due date has passed. A shortlist has been formed. Whether you're shortlisted, well and good. If you're not shortlisted, they will send you an apology letter saying that they're sorry, your application has not been successful. So there is a very structured interview process as well here. They will do first interview, which will be shorter. Then they will do a second interview, which sometimes will be on site. So they might invite you to come here and do an interview, meet the team, spend the whole day, see whether this is something you really want to spend your 3 years of your life doing, because it's like a make or break situation. Those important first years of postdocs are very important in setting the scene for you to become a successful academic faculty or a scientist in industry, for example. So yes, please do apply on most of the jobs in the UK are published on this website. Are there any questions, or shall we start? No problem. Okay. I can start the omics session today, and I promise I'll keep it brief because I know you have an overdose of omics by myself, and more importantly, very excellent talk by Dr. Aziz. So let's start this session. I want you to ask me questions as you feel fit by typing them in the box. So this is a regular central dogma of molecular biology. DNA makes RNA as a result of transcription. RNA, not all RNA become protein. As we know, there are lots of RNA. In fact, majority of them, different types, which never make protein. Only the mRNA make protein. You know that, right? And many of the proteins, we know most of the enzymes are nothing but proteins, and they are involved in cellular metabolic pathways, and they can lead to formation of new metabolites as a result. So sometimes through metabolic pathways, you can get hints of which proteins are active in a cell. So studying them, looking at P53, looking at ATM, I was telling you ATM phosphorylates P53, well, good. But I also told you that ATM also phosphorylates 800 odd other proteins in a cell. So there are a number of substrates of ATM kinase in a cell. P53 is just one of them. To make all of this sense altogether at a system level requires lots of skills like RNA sequencing, mass spectrometry, which I believe Dr. Aziz discussed with you previously today. So transcriptomics is nothing but to measure the level of the mRNA of different genes at once in a cell. That is what we study when we study transcriptomics. The way to study is through either microarray, which are still in use but have been phased out mostly and replaced by a technique called RNA sequencing. Then you have proteomics, which is something my lab currently focuses a lot on studying proteomics to measure the abundance of proteins, because ultimately proteins are the ones which are the drivers of cancer, and more so because proteins are the ones which are comparatively easier to target compared to RNA or DNA. Of course, there are lots of non-coding RNAs, microRNAs that people are trying to target, but the one which has been most successful in targeted therapy is to target proteins, and the proteins that cause cancer in industry and hospitals, of course, clinical systems. Metabolomics is nothing but to study the metabolite, what metabolites are being formed by a cell, what types are they. So ohm is a simple word that you can use to define an entire set of proteins. So gene, you can say it's a genome. Protein, you just said proteome, it becomes the entire set. So that is what is defined by ohm, and the study of it is called omics. I'll keep it short. So you can apply omics to define cellular pathway these days. For example, have a look here. This is a TCA cycle or Krebs cycle as we know it. There are different enzymes of the cycle which will become active when the substrates are present. So you can, with the help of the transcriptomics, proteomics technology, you can work out all the mRNAs which are overexpressed in cells in a specific stimulus conditions. These are the metabolite enzymes which the cell is making. These are the proteins, and these are the metabolites which are being formed. So what has happened with transcriptome, proteome, and such a discovery of these technologies is to become much easier to do discovery research. Um, but I think we are getting more intellectually weakened, especially with the advent of artificial intelligence and the reliance. There are reports that we are not learning as fast as we did before, thanks to the AI. So I would say still human intelligence, there is no substitute to human intelligence. Um, and You can see here in terms of this, to discover scientists, Hans Adolf Grabe, to describe this biochemical pathway, to see which enzymes are present, what are their cofactors when they are incubated with the substrate, several of them, what products are formed, must have taken not only their lifetimes, but also required a lot of intelligence. Now there are things which you can do using omics is to identify all of them in one go. I'll show you in an example. But within this Krebs cycle, you can see there is this enzyme called IDH. Mutations in IDH are very common. If this has not been discussed today, then I will certainly catch up on this tomorrow. You can see isocitrate dehydrogenase. The gene makes a transcript, then a protein is made, the isocitrate dehydrogenase protein, which can convert this isocitrate into a product called alpha ketoglutarate. So you can see that when isocitrate dehydrogenase gene becomes active, you would see that the cell will have higher levels of alpha ketoglutarate compared to isocitrate. You block this pathway, you will see that the isocitrate will start to accumulate in a cell. So these biochemical reactions are painstakingly difficult to carry out, understand, make sense of them, interpret the data, requires much higher level of intelligence to decipher these pathways. Compared to now, we have tools at our disposal like these omics technologies, which does do facilitate the discovery research. But I have to say again, there is no equivalence of human intelligence, why these pathways are active, what happens, and there are a lot of things to do with these pathways, simple biochemical pathways in cancer research, which I can show you tomorrow. So you can see here this pathway as I gave you example of, and have a look here what happens. Why is it important that we study genomics, transcriptomics? One of the things you can see on the slide is part of a small genome shown here. It contains two genes, gene A and B. So what you would expect normally is that you can gene A is more active in a cell, and when I say the is more active, because it is making more mRNA. And what you can think that if there is more mRNA, we do qPCR a lot of time, thinking that actually if you have more mRNA, you will have more protein. Meaning these are linear events. That's what we think, that more RNA simply means more protein. Actually, it is not true for majority of the human genes. That not a lot of RNA. Which you can detect using qPCR technique, would translate into a lot of protein. Sometimes it is actually reverse, and I can give you some examples if you would like. And for example, gene B, it is not very active. When I say it's not very active, you must have seen from Dr. Hifzur Siddiqui's lecture today that genes which are not transcribed very well, they have epigenetic repression, they might make very little product in terms of mRNA, the transcript, and then it translates into very little, if at all, protein. But sometimes RNA is stabilized by different mechanisms in a cell, meaning despite having less RNA, you can have a lot of protein made by cells. And that's why I'm saying transcription and translation are not linear events in eukaryotes. In fact, even in prokaryotes, the same thing exists. But sometimes you may have a lot of RNA and it may not be translated into a viable protein abundant product. And that's why it is very important that we study the gene activation, what is called epigenomics, what we discuss, I think my colleague must have discussed today. And then we discuss, we work out the levels, the abundance of transcripts of all genes using transcriptomics, and eventually what proteins and in what level they are being made by a cell. That is very important. So how do we establish this? I think my colleague must have discussed these, Dr. Aziz must have discussed this today in transcriptomics. So I will keep it short. Dr. Aziz, did you discuss these? Microarray and so on?
Yes, I did. I covered it in the
You have.
morning.
Okay.
And
So I will
qPCR, I can give that a heads up as well tomorrow. But
Okay.
qPCR, I haven't
Okay.
told them in detail.
Okay, so I will skip this then. And you can see here, I will come to this example here. Have a look what is happening here. You have, is that what you covered as well, Dr. Aziz? Sorry, microarray.
Yeah, I had a very similar slide.
Okay,
So
I will
towards
skip
the
this then.
color.
I will skip that then.
Yeah, you can
And
go
I'll
ahead.
go
This will
straight.
be a revision for them as well. So I think it's okay. Probably they
Okay,
didn't get it in the morning, so they can get it now.
okay, I can tell them one story about transcripts of these six transcripts shown on your screen. So lots of proteins change in a cell. When you take any drug, you take out, let alone drugs, you take out cells from incubator and do a simple transcriptomic analysis. You'll find a lot of genes have actually changed in their expression. So it is a very dynamic process, and that's why what is happening in a body is sometimes, not sometimes, all the time, very difficult and different from what is happening in an incubator. And that's why we cannot really recapitulate what is happening to cells in terms of expressing specific transcripts. Is this the same? Is this being honestly recapitulated in laboratory conditions? The answer is not really. And that's why many times we fail. As many as 95, 99% of the molecules would fail in phase 1 clinical trials, because there was not very strong science in discovery science, first of all, and many times because these cells don't cry when you give them drugs. Of course, you can look at some markers, but humans, if they experience any side effects of these drugs, one of the most common one is neutropenia, so decrease in neutrophil, which cell type? Neutropenia, I can't remember on top of my head. So specific decrease in lymphocytes, I think neutrophils. That happens in many cancers. You're not going to stop any clinical trial for that. But if a clinical trial leads to a death, which is defined, on a drug caused by a death, not in an accident, going in a car, then this would be a serious challenge for a clinical trialist to continue with the trial that will kill a clinical trial. So in terms of gene expression, it is not linear, and genes actually, as Dr. Aziz was saying, they're part of networks. They are regulated, and they regulate sometimes as a pathway. There are certain genes which sit upstream of these pathways. I was showing you examples of androgen receptor, which was regulating some genes downstream by binding to DNA. I'll discuss this tomorrow. Have a look at this example. Somebody generated a knockout of gene C, and now they, from a cell line, you can imagine this is a cancer cell line, and that is, they did the transcriptome profiling, and that is what they get. So when they look at these 6 genes, A, B, C, D, E, F, that's what they found. They were part of the, they don't know what sort of regulation they have, but have a look here in green bars. This is how the transcript for gene A, B, C, D, E, and F look like. Now, in a bar from a different cell line in which the gene C has been knocked out through genome engineering, genome editing, see what happens. These cells do not make any mRNA for gene C, meaning your experiment was successful. And you don't see any, the gene is gone, of course the mRNA is gone, you don't make any mRNA. And you're seeing that this removal of gene C has no effect on gene A. It enhances the expression of gene B. It decreases the expression of gene D, E, and F. Can you start interpreting this data? What might be the role of gene C on these genes? Why do they look that they're changing the expression? You can instantly start to develop this idea as a biologist. I'm not talking as a computational biologist. I'm talking as a biologist. If I look at it, I would think, okay, it looks like this gene may not be relevant at all to this pathway, because if I block, if I get rid of C, there are no changes. I'm still making 100 copies of mRNA for gene A, sorry. And have a look here. Gene B has gone in expression much higher. It is making more copies in situations where the gene C has been deleted. Instantly, I should get an idea that gene C is actually causing a blockade of the expression of gene B somehow. So gene C appears, the product of gene C appears to be a repressor for gene B. Now, it may also be that this gene D, E, and F, 3 genes, they're decreased in expression when you get rid of gene C as a result of knockout. And you can see here the effect. All 3 genes go down in expression to some degree, 20-fold, 10-fold, 50-fold. And that gives you, again, something to think about. These genes may have been more reliant. For example, this must have been more reliant on the expression of gene C. Gene E was much more reliant than gene F. You only see 5 times drop in the expression from intact wild type cells to gene C knockout cells. There is a 5 times reduction in the expression of this transcript. Compare this with this, where there is a 15 times decrease in the expression of gene E when gene C is knocked out. So you can actually check the relative dependency of these genes on gene C here. So if I show you this pathway, you can actually start making this pathway. This is for your reference, the same as you saw on the previous slide. Have a look what is happening. These are 6 genes, and remember, gene A was totally unaltered. We think this may not have been part of the pathway. Uh, have a look what is happening here. It turns out, after biochemical experiments looking at interaction between the proteins, between the mRNA, that there are some interactions. What kind of interactions? Gene A drives the expression of gene C. Gene C drives the expression of these 3 genes, and gene E blocks the expression of gene B. So what is happening here is, if you get rid of this gene C, which we did in this knockout experiment, you can see gene C is totally gone. The mRNA is totally gone. When gene C is knocked out, you can see straight away gene D goes down in expression, so does gene F, and so does gene E. The effect, of course, was more pronounced on gene E, as you can see here, compared to gene D and gene F. This could have been one of many reasons, actually. But what is happening here, gene B now goes in expression. Why? Because gene E was suppressing the expression of gene B. By getting rid of gene C, what you've done is you have blocked the expression of gene E product, and that's why the gene B product now goes up, the mRNA, I mean. But you can see, I was telling you, based on this, that it looks like gene A is not really doing anything here, because gene A was totally the protein, sorry, the transcript, the mRNA was totally unaltered when gene C was present or absent. But actually, it looks like that gene A does regulate gene C because it sits upstream of gene C. So it is the major transcription factor, perhaps, which is regulating the expression. When you get rid of gene C, you can see here this upstream factor remains unaltered. So this is how you start to develop understanding of pathways. This is not something which you want to do with 20,000 transcripts of a cell, would you? And that's why we need smart, efficient computational biologists who can think out of the box to make sense of the data we get from a sequencer. Exactly. And they cannot really work alone. They need support from wet lab scientists who can tell them, start to make sense of this data, prove and validate the experiments that show that what I'm showing in a pathway are actually facts in which a pathway is regulated, being upregulated or downregulated by some components of a similar pathway. So you can certainly apply this knowledge on transcriptome and develop expression profiles. I can give you this example here. Have a look what is happening. There are the 6 genes. Each one is represented here by this line, and you can see here, this is the on y-axis. Sorry, there is no axis shown as such, but you can see this is telling you the expression level of these 6 genes. When you treat the cells with glucose and take out the mRNA, do the qPCR analysis, you find that when the cells are treated with glucose, all of them go up in expression. You might think that this might be actually glycolysis genes. They may be the ones regulating the glycolysis. But is this how the glycolysis pathway was discovered? The answer is no. It required efficient, smart, excellent biochemists doing painstaking research, bit by bit, each enzyme being expressed, not expressed, purified from liver extract, tissue extracts, and using those enzymes, incubating them with different substrate, not knowing what could be the substrate, and then looking at the products, what is formed, quantifying them, and then connecting these dots to create the pathway as we know, glycolysis. But nowadays, you can actually do this at astronomical scales. Have a look. This is an oversimplified example, glucose enhancing the expression. You use a different cell culture plate and treat this time with much higher levels of glucose, and you find similar things, that the expression of these genes is going up. Certainly, you will get an idea that these genes may be regulating similar pathway. So this is a metabolic pathway, but something similar actually happens in cancer cells, you might find that there are genes which follow similar expression profiles, which have similar expression patterns in cancer. And what we do as computational biologists, I'm not one of them, my colleagues are, who I collaborate with, that we develop these expression profiles. So what you're seeing on your screen is a map. Basically, it is a heat map. And what you are seeing in this heat map, each there are three individuals. So each column represents the expression of a particular gene in each individual versus in healthy males versus patients of prostate cancer. So now you might ask me this question. Perhaps many of you already know, who are advanced students in their academic ladder, is why would you get prostate tissue from healthy males? Well, whenever surgeons are doing surgeries, there is a principle of surgery. You don't leave anything behind. So you need to make sure that all the cancer has been taken out, has been excised, even at the cost of removing some nearby non-cancer part where you suspect there may be some cancer cells which are trying to invade through the organ. So the surgeons, if there is any surgeon on the call, they can elaborate in more detail. But the principle is, you don't leave any cancer cell behind, and you need to make sure that the margins of the disease, there is no positive margin left when you slice the tumor out from an organ. So that's why you will get this tissue. They will go to a pathologist and they will say, hang on a minute, yes, there is a tumor core. Most of these cells, they're all cancer. But when you start to cut the sides, the periphery of the tumors, you might realize that actually the margins are now clear. The tissue that you're getting is entirely normal tissue, meaning you have made sure that you've taken out all the tissue which is all clear now. And that actually is not thrown away, but actually you can isolate mRNA, do the RNA sequencing, and use those tissue either as benign tissue, or some people, in case of prostate cancer, will give out their tissue out of choice, because prostate gland, as we age, grows in size, and where it sits is underneath the bladder, as I mentioned, and it surrounds the urethra, meaning it is going to block the bladder being completely emptied. And as a result, these patients will go back to the toilet again, and they will still feel that their bladder is full. Those patients will undergo a procedure called radical prostatectomy. And that means their prostate gland will be completely removed. Those patients will have their studies, if they're part of any study, or if they give consent, then their RNA sequencing data can be, transcriptomic data can be obtained, and then compare with patients who have prostate cancer. Now, I want you to have a look here what is happening with the color. So the red color shows high expression of genes which are listed in these rows. This is one gene, gene A, for example, gene B, gene C, and so on. And the same expression in cancer conditions. So have a look here what is happening. These genes, set of genes, they are overexpressed in normal healthy males, but their expression has gone down in terms of mRNA expression has gone down in cancer. What do you think, what we covered in the last 2 hours, where would you categorize them? Are they oncogenes, or are they likely to be tumor suppressors? Their expression has gone down in prostate cancer patients. I would think that they're likely tumor suppressors, or genes which were keeping the organism safe from developing cancer. Have a look at the second subset here. What is happening? The expression of these genes, which are down regulated, as you can see, the color green means down regulation, low expression. They are actually overexpressed in cancer patients. 3 of them are shown in this example, and that can actually form a signature. So these days, cancer is treated, we have new therapies of cancer. Sometimes what people are looking at is at the specific signatures in cancer. What type of signatures are there? They can call them signature 1, 2, or 3, and based on this, patients can be put into specific clinical trials or a specific drug regimen based on the expression of particular phenotype. Imagine someone who does not have any HER2 overexpression being given a drug Herceptin. Would it be effective? No, it might have side effects. So this is certainly very important, and we can make these heat maps. This is one of the oversimplified heat maps. If you go and look at some of the literature on cancer genomics, clinical cancer genomics data, you will find that these are enormous maps can be generated for hundreds of patients from one or multiple trial centers, and genes can be mapped, and such plots can be formed. And from this, you can deduce, you can get the signatures out of genes which are possibly the ones associated with cancer. So these are the ones, if you take them out and see what these genes might be, you might find that actually these are the ones, some of which are already known to cause cancer, some of which are already known to be oncogenes. So if I give you this example with the regards to the molecular subtyping, you can see here uh, there is something important which is called molecular subtyping. What you're seeing is called the IHC based data. This is how the patients, women who have breast cancer, are treated their cancer by pathologists in the lab, and this happens in every country, including India. Their cancer can be defined as having expression of estrogen receptor, progesterone receptor, or her receptor. This is called, this will be, if they have all three of them, we will call them triple positive. We don't really call them that, but we call them triple negative. Why? Because if you are triple negative, as we discussed this morning, morning UK time, triple negative actually have very poor prognosis, meaning the outcome of the therapy treatment is really, really bad. I showed you those graphs, Kaplan-Meier plots, and triple negative patients who have none of these actually do very badly. These tumors, pathologists can tell you these cells look like basal-like. They have specific cytokeratin markers which will allow them to categorize them as being basal-like. If you're luminal-like, then the chances are that you will respond very well to the therapy. And have a look, something else is happening here. If I'm giving you these two examples, basal and luminal-like, have a look at this basal-like says key 67 negative. Now, those of you who don't know key 67, it is a marker for proliferation. Any lab studying cancer, any clinic, any hospital doing biopsies, they will look at the proliferative index, how fast the tumor is growing. You have more key 67, of course, the chances are that the tumor is fast proliferating. Many times it is a bad news, but have a look here. In this case here, where the prognosis, where the outcome of the disease is super bad, estrogen, progesterone, HER2, all three are missing, but key 67 is also negative. They're not growing very fast. So does that mean that if you don't grow fast, you're not cancer? Well, it is cancer. It is not growing fast for now. It has probably gone into a state we call as senescent. In P53, I was showing you a slide. Those cells are still alive. They are just waiting out for the right time when the drug will not be put on them. There will be no systemic or other treatments so that they can actually get revived again. Have a look here. These cells don't divide. Many of the cancer therapies, especially chemotherapy, will depend on the ability of cancer cells to divide at a faster rate. You divide faster, you die faster as well. But in this case, these cells are not very much in a dividing phase, meaning any chemotherapy you throw at them is likely to be unsuccessful. So why am I telling you this in this omics is because at the moment, what the signatures we have, they work. We have only of these few handful of subtypes, 1, 2, 3, 4, 5 shown on this screen. And you can roughly divide them into good prognosis, women who will do well and probably will have become cancer free, versus those who are likely to do bad. Sorry. So what you can appreciate is that with omics, we may be able to make more precise subtypes and identify these signatures. In this case, this is shown for prostate, but there are plenty of data. Have a look at this data for breast cancer, which is nothing but came out from omics of more than 1,000 women. It's called MetaBrick data from Cambridge University. And that has a lot of data. I've typed it in the chat box, MetaBrick data for the clinical genomics of women with breast cancer. They have exome sequencing data, they have transcriptomics data as well. So coming to the last slide before I start showing you that platform, let me know if something is not clear. I can certainly uh, show you again uh, those slides, but a lot of what we are going to discuss is going to be based and relied on your understanding of this last slide, which is here. Have a look what is happening here. This is super cool. It looks excellent, and this is exactly what you're seeing is much more complex data, but similar data what I was showing you in this chart here. So what you are seeing here is the expression of androgen receptor. Why androgen receptor? Well, this is what I study for past almost more than 2 decades now. So you have patient samples, the RNA isolated, the RNA sequencing done. So what you can do is you can isolate mRNA and you can do RNA sequencing, and you can see there are more than 500 patients. You can see this scale. This is 50 samples. And what you can do as a computational biologist, you can make a spectrum of this where you can put these patients who have either very high expression of androgen receptor and patients who have very low levels of androgen receptor. Now you will tell me, hang on a minute, you were saying androgen receptor drives prostate cancer. Yes, it does. But like breast cancer, where I told you estrogen receptor drives breast cancer, HER2 drive cancer. We also have breast cancer phenotypes like TNBC, triple negative breast cancer, which we don't know what is the major driver of TNBC. And there is an intense research investigation to identify the drivers of TNBC. Likewise, here, there are prostate cancers which do not have any androgen receptor. And you can see within this data, these are about 70 patients who are negative or they have very low transcript levels of androgen receptor. Now, have a look at the second bar. So this allows you totally free without having a login, this Xena clinical genomics data set to allow you to study your own data as well as study the data which has been submitted as a part of this consortium, TCGA consortium. You can see the level of number of samples, over 500 only for prostate. Your favorite cancer, have a look there. You will find it in that data set. And we will, in the second half, I'm going to stop soon. We will, in the second half of this tutorial, we will have a look at this platform. Have a look. This is a second gene called ARID1A. This is just not a random gene, but we know ARID1A is one of those genes which bind to androgen receptor protein. So protein binding with protein and regulating, promoting the function of androgen receptor. I should tell you a lot of these slides, and I did mention that what I was showing you was oversimplification. Many times proteins work in coordination. They bind to each other to drive specific activities. We saw with cyclin D, there were 3 proteins binding to it, but chances are that there are other hundreds of proteins binding to that complex, which we don't know yet, or somebody might have worked out. Androgen receptor, we have recently found binds to about 1100, so 1100 proteins are now known. We have established that bind to androgen receptor. Some of them were already known through biochemical experiments, through yeast-2 hybrids, mammalian hybrid systems used in 1990s. But now we know that actually there are lots of proteins which we never knew can bind to other proteins, especially the drivers. So in this case, ARID1A, you can see here, patients who have very high levels of androgen receptors, it seems they have very, very high levels of ARID1A. Conversely, patients who are very, very low in androgen receptor, they don't need ARID1A because ARID1A is needed to drive the activity of the androgen receptor. So they're very, very low in androgen receptor, they're very, very low in ARID1A. Makes perfect sense. Have a look at this third gene, A1BG. And A1BG is not something that we study in prostate cancer, but having high levels of A1BG is a good prognostic marker in liver cancer. Meaning, if you have lots of expression shown in red, those patients in liver cancer will do better. They will live longer compared to those patients who have low levels of A1BG. So you can think that A1BG may be a tumor suppressor. Who knows? So you can see here that what looks like here that there is an opposite correlation. Patients who have very low levels of androgen receptor have very high levels of A1BG, meaning it is possible that A1BG is a tumor suppressor even in this context. And androgen receptor was inhibiting the expression of A1BG. In tumors who don't have the androgen receptor, A1BG cannot be expressed. So you can see how complex these data sets are, and they can be used, patient data, to make real world discoveries thanks to platforms like Xena, clinical genomics platform, which I will discuss with you in about 10-15 minutes from now, and show you what we can do with the help of these platforms, clinical genomics studies, to study the transcript expression and see how the transcript for gene A, for example, correlate with gene B and gene C, and so on. So I would summarize this. I'm not going to read this out, but basically this is in a nutshell what we have covered so far in this omics tutorial. And with this, I will stop sharing my screen and go to the Xena platform after 10-15 minutes, unless you have any questions. Shall I stop sharing my screen? Dr. Aziz, shall we
I
take it?
think your lecture was very complimentary to mine because you went in depth
That's great.
and I skimmed the surface. Uh,
That's
I
very
think
good. That
nice
compliments
summary.
very well. Thank you.
Although I told them that microarrays are obsolete, because I did that myself and then I no longer use it. But yeah,
No, that's true.
keeping
Microarrays are not used. They're used for a specific purposes. For example, focus custom arrays for proteins. Immune arrays, more like they are not used these days. You're absolutely right. We use mostly RNA sequencing these days.
yes.
So shall we reconvene in 13 minutes from now?
Okay.
And start, and or if you want, we can crack on and finish early, because I realize I'm only starting my day and it's the end of the day for most of you. So if you want, I can continue, or I can stop.
Well,
Your
that's
choice.
totally up to you. I think.
I'm fine with anything.
Okay. Then I think let's continue because
Okay.
taking break might drop some
Yeah.
of the candidates here. So
Okay, that's fine. I will stop sharing my screen, this presentation, and then I'll start presenting some of the platforms I'll show you today. Okay. So let me just start this. So first thing first, how would you know whether there are genes which are important for growth or not? Um, is there anyone who can think? Is there any platform? So I will go to Xena as well. But does anyone know any platforms that they can use to find out, okay, this is the cancer I'm talking about. This is GeneX. This is part of my PhD project. Uh, Before I start a knockout experiment, how would you know that this is actually the driver or potential driver, possibly? And there are lots of information available on the web these days, supported by some excellent institutions around the world, where you can get such information. And let me show you my screen. How would you find that data? Dr. Aziz, did you cover that in your lecture?
You're talking about UCSC Jena, right?
No, can you see my screen?
Yes, cancer dependency map. Okay. I thought you were
Yeah,
talking about
did you show it to them?
no, no depth map minor, no cell model passport, no. So this all would
Okay,
be new for them.
great. I think we can access this here, or let me check. It would be on
Can you
Sanger.
put the web link for this?
Sanger.
Depth map.
That
Okay, for the cell lines.
That
So
map. Yeah, I think I'll go to.
you can type
This
the
is
web
really
address.
really cool. What do you can? Uh, Dr. Aziz, I think. So this is not something you've discussed. I think when it was started, you did attend the conference. Uh, you were once in Cambridge.
Yes, yes, yes. Hingston.
Yeah, I think so. The depth map is Hingston, yeah, it's a village near
Yeah, we use
Cambridge.
this DepMap portal
Mm,
quite often. In
Don't
fact,
think
one of my
bad about
PhD
villages.
students who has
That's a
worked
big
on
thing.
DepMap.
Oh, you do use.
Yes.
Oh, you do use it. Okay. So
Yeah.
DepMap is an excellent platform to see what happens to the expression of your own genes. For example, if you have a project, you're working on it, and
Can you please,
as part of
sorry,
PhD, a
sorry
master's
to interrupt.
project,
Can you just type the address, web
please.
address on the message box so that if users want to
Okay,
follow?
let me do that. Yeah. So, before you start spending time doing research, i- for example, making those hairpins, devising a knockout protocol or a siRNA protocol, and start inhibiting this protein in a cell based on some data that shows that this protein is overexpressed, you might as well actually look into this platform and see what is the impact on cancer cells of getting rid of removing through CRISPR this particular gene. I've provided the link. I will type in the name of my favorite gene unless somebody else has anything else to tell me. Uh, Androgen receptor. Not the agonist. Let me type in AR. So androgen receptor, if you type in the symbol, it comes up. Now have a look here what is happening in these data set. First and foremost, you can see the location. I was telling you androgen receptor gene is located on chromosome X. These are some of the names, alternative symbols for the androgen receptor. What I want you, there are lots of information here, expression data, copy number data, mutational data, dependency data, structure, inhibitors. So you can actually pretty much uh, write a BSc thesis, at least if you study this more extensively. What I want you to do is see here, this is a perturbation effect. Perturbation simply in this term means that you're going to inhibit androgen receptor uh, gene expression and measuring the effect on cell growth. Have a look what happens here. So if I click here, that's what you're going to find. So this is 0, and then you're seeing there are different cancer types here, and even within different cancer types, if you go to prostate, you will find here that this is the cutoff here. And you can see in these two cell types, LNCAP 22, these are cell models. You know there are different models of different cancer, which you can grow in an incubator in a lab. And you can see here, by getting rid of androgen receptor in this 22 RNA1 cells, and by getting rid of androgen receptor in LNCAP cells, you can actually diminish their growth. So these are the two cells where getting rid of androgen receptor would mean that their uh, proliferation will be badly affected. And rightly so, because we know that both of these genes, LNCAP and 22, both of these cell lines express very high levels of androgen receptor, and they are dependent on androgen receptor. That's why we call it dependency map or DEP map. Uh, as opposed to others, you can see there are very little effect whatsoever on other cell types. Does that make androgen receptor a good target? Yes. Why? Because if you're going to make therapies, if you're going to synthesize drugs that block androgen receptor, they're going to have a very bad impact on prostate epithelial cells, cancer cells, but they will not have very much side effects on other tumor types which do not express androgen receptor. So you can see getting rid of androgen receptor in these cell types, ampulla of Vater, biliary duct, bladder, bone, does not really have much impact. So you can get some first-hand evidence of the impact that the knockout perturbation of particular gene A, B, or C will have on your cancer type, or even normal cells, for example, eye cells, fibroblasts, head and neck, kidney. By the way, these are mostly the cancer examples like head and neck. You have some data, how many 77 data sets within this. So this is how you're going to do this analysis and identify first hints. I'm not saying if this data looks negative for your protein, don't go for it. What I'm saying is it will give you a good idea to do some sanity checks and to reflect whether the gene that you want to study as part of your PhD in particular cancer type, is it likely to have any relevance there, or this is completely out of question. This is something you can get by studying it from using the DepMap platform. Any questions on this? So what we are developing at the moment, you can see here, this platform allows you to study only one gene in a different cancer types. Because my lab studies mostly prostate cancer, we have downloaded all the data which is submitted to DepMap portal. And thanks to my computational biology colleagues who've done this for us, we are able to study any number of genes in cancer and the effect they have. If we want to study 200, get some idea, there are 200 genes. There seems to be a signature that I have been asked to study. 200 genes, that means you sit all day long and you want to study on prostate, and that would mean it will take a lot of time and you will not get comparative data, meaning getting rid of androgen receptor, kills these cells, and you can see the gene effect is shown in -1.4, expression 7.59. So very high expression, and gene effect is very, very high. These are log values. You can see very high expression, and getting rid of androgen receptor would kill those cells. But this is only for androgen receptor. If you were to do it for 2,000 genes, for example, 200 genes, it will take a long time. So what we have done is developed a platform that allows us to do the same, but do that only for prostate, but for multiple genes. This is still under development, so I won't be able to show that to you. That's one thing. So that gives you first idea of your gene you want to study. I wish you would have told me a gene if it was not a secret. Then I could have shown it to you. One of you could have shown, but I understand you can do the same analysis yourself because this is being recorded. So Padmavati is asking, are those maps based on experimental studies? 100%. So these are all based on, I should have mentioned this, this is all based on CRISPR screen, knockout screen of CRISPR. And you can see here, this is the CRISPR knockout screen that is showing here that we are using. So this is the CRISPR DepMap publicly available data set that we are using, Padmavati, in this. So for example, androgen receptor sounds very good. If you want to study in,
is there
yes,
any
please.
way that we can know about the off-target effects, if there are or there are not?
off-target effects of the expression. I think what you would want to do, so this is an excellent question, Dr. Aziz. With this, you get, because this is a screening data, it simply means it has not been validated in vitro. So I will take this data with a pinch of salt.
Yeah.
Th- this is not something you can take at face value. You have to do multiple other testings to make sure that no, this is not something you would want to study. For example, androgen receptor is expressed in breast cancer cells as well, and you can see gene effect. It is showing minus, meaning in breast cancer cells also getting rid of androgen receptor affects their growth, but also androgen receptor is expressed in liver cells. So let's see where the liver is. Have a look at this liver. This is again showing very little but negative effect of androgen receptor. Getting rid of androgen receptor decreases their growth, it seems. So there is positive expression. These cells are expressing androgen receptor. We know liver does express androgen receptor, and one of the ways people have tried to stop hepatocellular carcinoma growth is by giving androgen receptor inhibitors. And to some extent, it has been successful as well. But the prominent role of androgen receptor is only so far in prostate cancer and molecular apocrine breast cancer. So that's an excellent question, Dr. Aziz. You have to see what sort of data you can get. And again, the question of side effects will depend on whereas this protein is being expressed. Are they being reliant on this protein as well in terms of their growth? I hope that makes sense. So
Absolutely, yes.
thank you. So I'm going to show you another platform now, which is very good to see. Okay, there is protein which is being expressed in this particular cancer type. Remember, the data you're seeing on your screen is taken from cell lines models. This is not data taken out from real tumors. So if you want to have a look at the expression of androgen receptor in real tumors, is there any way that you can get that data which is publicly available? Yes, you can. And there is a platform which is called Protein Atlas. If you can please, I will provide the link here. Human Protein Atlas. So if we go to this platform, can you please confirm that you can see this page uploaded on? Thank you,
Yes,
Swati. So Protein Atlas is an excellent, thank you, Dr. Aziz. It is an excellent platform as well in terms of studying the primarily I use it, my lab uses it to study whether any new proteins that we get from screen mRNA, are they even expressed at protein level? I told you that protein expression and mRNA expression are not linear, meaning if you see a lot of mRNA, it does not mean that you will have a lot of protein. So before you start doing that journey, and I understand there are inhibitions, antibodies are super costly in India. qPCRs, getting primers is, of course, much more feasible and easier. It takes time from what I hear from colleagues in India. So you can actually do a lot of discovery research using this platform, preliminary research for your own project. And you can get actually, let me show you. So we use it to see whether proteins that we find in the screen, they're expressed, are they expressed in cancer. So for example, if I go to androgen receptor, you can see here in this platform, it has recognized that there is androgen receptor. You click on it and it will give you a lot of data. Now they have combined it so that there are lots of other data from tissues. There are interaction data. Remember what Dr. Aziz was telling you, pathway analysis, and there are interactions between proteins. That is what it is talking about. The three-dimensional structure of the protein and so on. I want you to have a look what happens in the real tumors, whether they do express androgen receptor or not. You can study and treat cells in cell culture. You can kill them, but is your protein in fact being expressed in cancer or not to have any clinical relevance in the first place? This is something you can do by using this platform. So if we go and click on cancer, uh, it looks like this is showing you the TCGA, the same data set I showed you previously in Xena browser. I'm going to show you the Xena browser in a minute. Have a look here. It looks like androgen receptor is highly expressed in some forms of brain cancer. It is expressed in some forms of prostate cancer. Also in breast cancer, androgen receptor expression, transcript expression at least, is very high. So if I go on prostate and show you some slides, in fact, that is what you can access from this platform. So if I go to prostate, you can see immunohistological, immunohistochemistry data from these slides. Have a look here. They have patient data, core by core, provided for patients stained with androgen receptor, and see the expression level yourself. So you have to be a pathologist to understand these slides. I'm not one of them, but I can tell you based on my experience and what I'm seeing here, these are the glands that are the ones that give rise to cancer. These are the glands within the prostate tissue. What you're seeing here is a stroma or connective tissue. That's what we discussed before, where the connective tissue is. I was showing you a cartoon, and these are the glands, some of which are going to make cancer. You can see the cells expressing very high levels of androgen receptor shown in this brown stain. Uh, it says very high staining, and you can see if you were to do staining to do the nuclear stain, you would see that the staining will, nuclear staining such as DAPI, will tell you that these oval blocks are actually nucleus. What you're seeing here, can you see my cursor?
Yes, sir.
Thank you, Swati. So what you're seeing here is actually extracellular matrix, but also, the lumens, the ducts, that's where the secretions are going from these cells. But also nearby, what you're seeing is a cytosol, which is not very visible in this picture. So what you see is that androgen receptor expression is largely restricted to the nucleus. That's what we are being told by this platform. And you can see here, you need to be a pathologist to identify whether this is cancer or not. In this core, you can see there are some cores which look like they have lost the structure. So normally you will have a two-cell layer. One of them is the luminal cells facing the luminal. The other one sitting on top will be the basal cells. And that is the architecture which is abrupted, which is gone in prostate cancer. So I would say this is prostate cancer. And moreover, what I can say for confidence, that indeed, yes, this protein that I want to study and spend next 3-4 years of my life doing a PhD does indeed express androgen receptor. This is androgen receptor, which is very well known. You might apply this knowledge to study a new protein you want to study in order to have its clinical relevance identified, deconvoluted during your PhD. And that means you would know that what you're doing may one day actually have a clinical impact, because the protein that you're studying and trying to block using understanding the pathway that actually activate this protein may actually have one day effect on the clinical treatment of the disease. So that is something I extensively allow and encourage people to use to have a look at the expression patterns and of your protein of interest. What it also does, it tells you some antibodies that will definitely work in IHC protocols. So they do sell some antibodies. They do recommend some antibodies, not directly the platform, but they will tell you mostly from Sigma where you can get some antibodies which will work very well in identifying cells which express your protein of interest, in this case, which is androgen receptor. So I hope that is clear. I can go back. You can see there are different cores from different patients. Of course, there are no patient names, but what you can see here is that they have been coded. For example, have a look at this. All you see is mostly stroma, but you see there are some normal and cancer cells, cancer epithelial cells as well in this slide. So that is something very good that I would recommend, highly recommend you use. And the last platform for today that I'll talk about is likely to be Xena browser. And let's see what we get from it.
I've
Information.
already
So what I
posted
would
the
try
link.
to do is to replicate what I will discuss. Sorry,
I've already posted the link there,
Dr.
but it's
Aziz, you were
showing insecure.
for Xena. Yeah, okay. Let me do that. See if you can access this through this platform, xenabrowser.net. So this is the ultimate platform we have to do some discovery research on clinical cancer genomics. It may sound like slightly complicated, but I've tried before with my omics tutorial to make sense of it and make it easier for you to understand. So first of all, this is a platform where patients, the samples, especially the transcriptomes, have been submitted, and the expression has been arranged into high expressors versus low expressors, and you can do it at a single click based on any protein or group of proteins you want to study, and you want to see what is their correlation with each other. So I was giving you an example where I was showing you the androgen receptor expression of ARID1A, the protein that binds to androgen receptor, and I was showing you that if you have low androgen receptor, you have low ARID1A. But there was another gene, A1BG, which seems like follows the opposite pattern. So first, what we will try is to replicate those findings using this platform. So let's carry on. So first of all, that study was prostate cancer. So if you go here and select this study, I hope you all can see that. What you can do is select that study that was PRAD. PRAD stands for prostate adenocarcinoma, or short for prostate cancer. So you can select this study here and use it. This is the study. You can tell the system, I want to study. I want to see what is happening with the gene that I want to study during my PhD, for example. So you can see here, there are still 568 samples, the number of samples in this data set. And what you want to study is the level of mRNA for androgen receptor. And you can tell the system which database do you want to study. And to start with, you can simply say, I just want to see gene expression, meaning what happens to mRNA of androgen receptor in patients. If you do that, this is going to, again, put these patients into high expressors. You can see these patients have very high level of androgen receptor transcripts. These patients on the other side of the spectrum have very low level of androgen receptor. And now you've been given a PhD project where you want to see your gene of interest expression with, let's say, androgen receptor, and compare it with androgen receptor. All you have to do is type in the name of your gene. So A1 BG, as you can see, stands out here. That's why I used it as one of the genes. But I'm going to show you the gene which is known to be a partner for androgen receptor and regulates androgen receptor activity. And that will show you that actually these are part of the same pathway. ARID1A gene, and we are again going to do basic analysis, gene expression, and click done. Have a look here. This is one of the finest overlap you will get. So you can also zoom in, and if you want to zoom in, just hold your browser and click here, and it is going to zoom in your data. So these are, now you can see the scale has shifted. Previously it was 50 samples. Now it is telling you these are 5 samples. So you can see very neatly patients who have very high level of androgen receptor across this board likely have very high levels of ARID1A. Why this is important? Because we understand we cannot target androgen receptor in prostate cancer. It becomes mutated, it becomes amplified. Fair enough. We know that genes change, gets mutated in cancer. But perhaps we can target what makes androgen receptor active. In other words, ARID1A. And in fact, people have done this. So they try and target ARID1A using drugs that will block ARID1A function. It is noteworthy. It is not important only in prostate, but it is also important in driving some forms of breast cancer through estrogen receptor, because androgen receptor, estrogen receptor, they're very analogous in terms of their activity. They're both nuclear receptors, and it is known that ARID1A can support prostate breast cancer growth as well by binding to estrogen receptor. So you can ask this question: is this the only gene which is highly expressed uh, or co-expressed with androgen receptor? There is a separate platform. If there will be interest, I can show you that platform today to see, okay, my gene is in cancer is gene A. I want to see this. We already know that ARID1A is overexpressed in cancer because it is a partner of androgen receptor. How about that gene which shows the maximum activation or maximum follows the same pattern of expression as androgen receptor? ARID1A is one of them, but is it the top 5 genes, top 10 genes? There is a separate platform that will allow you to do that. So this is what we were showing. I showed you in the omics tutorial. You can zoom out again or clear the zoom. So if I clear zoom and type in the name A1BG, let's see what you get. That was the name I showed you. This is a good prognostic marker for liver cancer, and study the expression pattern of A1BG. You should be able to essentially replicate what we were getting before. Yes, that patients who have very low levels of androgen receptor have very high levels. I can zoom in and show it to you. Have very high levels of A1BG. Have a look here. Very low levels of androgen receptor, very low levels of ARID1A, but very high levels. Most of the patients have high levels of A1BG. That should suddenly tell you something. You can start uh, deriving some conclusions based on this. Perhaps androgen receptor, as I was telling you, is repressing the expression of A1BG. And you might want to go back PubMed and check whether there are reports of A1BG being regulated by androgen receptor, where androgen receptor serves as a repressor for this A1BG gene, which we know in liver cancer is actually a very good gene. High expression protects against liver cancer. Perhaps this might be similar case in prostate cancer. Who knows? Because androgen receptor blocks it. And we know androgen receptor promotes prostate cancer. So if you connect all these dots, it may be that androgen receptor promotes prostate cancer by blocking A1BG gene. So you can actually get a lot of data, very good publication quality data, or at least to start with your PhD, a lot of data for your synopsis to think about what you want to study in during your PhD. So I'll clear the zoom and stop this here, the Xena browser. You can see ARID1A is one of the genes which is highly expressed, shall I say co-expressed, and follows the same expression pattern as the androgen receptor. Dr. Aziz, will I have 10-15 minutes to take questions, but also to show them a platform which will allow them to find out which genes are the most expressed, top 10 genes which are most expressed with their gene of interest. Uh, ARID1A, I knew it, so I showed it to them, but that platform will tell them actually there are maybe part of same pathways and follow the same expression patterns. Okay, so let me take questions first. I'll take some questions first. Are there any questions? Please make sure your mic active and ask me any questions. Padmavati is asking, can we use data from these sites for publishing papers like meta-analysis? Yes, 100% you can do that. There are specific journals which will take such data and publish. You can publish it. Normally, for any good publications, may I suggest you need some elements of in vitro validation, some molecular biology validations through some experimentations to publish them, because right now there are parts of the world where this is becoming quite common. People have developed tools to bring all of these platforms on a single portal where they can access and uh, basically churn out a lot of data in no time. And as a result, there are concerns that what is the meaning of these data if it is not validated through experiments, and therefore there are journals which will decline to publish anything which is 100% computational biology. Fair enough. But yes, there are journals who still and very well welcome data. They're low impact journals, but they do take data only as computational analysis for publication, because they are publicly available. If you do your own analysis, like I showed you the paper yesterday, the mutation paper, the air pollution paper, nature paper, most of it was basically computational data from patients, but that was not publicly available. And all this those authors did was to analyze it. No, that was their own data, and they took time to do lots of extensive analysis, uh, showing some signatures coming out of that data and publish it. So yes, if you have your own computational biology data, that will certainly lead to very high impact papers if very relevant, and of course it depends on the journal as well. I hope that answers the question. Any other questions? I can show you then a platform. That will be the last platform. I'll come back again to showing you the Xena in a minute. But I want you to have a look at this platform called, I'll keep this like this. It's called GPR2 platform. So I will give you the link in the chat box. Okay. So in this, looks like it is not working for some reason. Okay, I don't think this is working, this platform. So you can study a lot of your proteins of interest using this platform. I was telling you that androgen receptor interacts with this ARID1A, and a lot of proteins, in fact, interact with a lot of other proteins. There are actually interaction maps. Dr. Aziz was showing you some examples. There is a platform where you can actually access publicly available data sets like the proteomics data sets coming from mass spectrometry. And one of the platform is called the protein uh, string data set. Have a look at this in your own time. This is an excellent data set to tell you the protein that you're going to study during your PhD, your MTech, your Masters. What are the binding partners of your protein of interest? This is an excellent platform, and I would suggest that you have a look at this platform. Uh, so you can see here, this is a database of all the protein-protein interactions. They may be physical interactions. It may be one protein regulating the expression of other, or vice versa. This is what you're seeing on your screen. And what is happening here is that this is the data for over 12,000 organisms. How many proteins? interactions are there? Over 20 billion. And how many proteins? 59 million proteins. So you can see how diverse the protein numbers are in different organisms. They may be paralogs, they may be proteins which are alternatively spliced, or altogether they may be totally new proteins, who knows. But you can actually identify, when you start a project, you can identify proteins that interact with your protein of interest. All you have to do simply is click this button, search. And if you go to the protein name, you can type in the name. For example, if I type in a wrong name, such as androgen receptor, it will not identify anything, meaning there isn't anything like that. So you need to identify the correct name. Oh, it's still not showing. So let's try that. And Homo sapiens, which we are, and type in search. Let's see if it finds something. Yes, it has found something. This is what we are talking about, androgen receptor. We are not talking about any other thing. So if you go and this is the one that you want to study, you can continue. If this is not the protein that you want to see, what is binding to my protein of interest, you might go and have a look at other proteins of your interest if they are the ones there. So if we go to the androgen receptor in Homo sapiens, we are not talking about any other species, and continue. Let's see what you find. I believe this is not something which you have been discussed. Uh, and have a look. These are the proteins that it is showing. Androgen receptor is known to bind. So you can play around with the binding. You can make this extended, see what are the core binding partners of androgen receptor, but take this with the pinch of salt, as I mentioned. This is the data, which is what we have found. Androgen receptor is to interact with 1100 other proteins. This is only a few proteins that the data is showing, but this is a rather still quite important, because it tells you that these are the proteins that interact with your protein of interest. If you're someone who is doing proteomics studies, you can certainly use the this link. You can register and log in to upload your own data set to study them through the platform, to see whether there are any patterns of interactions. For example, here, these are different nodes that tell you what is happening. One of the predicted partners, like I was showing you with Xena, what was binding to the androgen receptor, ARID1A, was following the same patterns of expression as the androgen receptor. In this case, this data will tell you what is the predicted functional partner, meaning the protein that is likely binding to the androgen receptor. Have a look at the first one, HSP90, which is nothing else but a heat shock protein or a chaperone. What is the score the system is predicting for the binding with androgen receptor? It is saying 0.999%, 999. And that means this is the most score you can get, is 1.0. You don't see it, but normally this platforms give you the score if it is high binder as 0.999, which tells you this is very likely a protein that binds to the protein of interest, my protein of interest. And indeed, this is the case. Chaperones, HSP90 family chaperones, they bind to androgen receptor and stabilize it. It is very well known. And then you can actually study what is the evidence for this. And it says there are experiments done. So what it is doing, this platform, is curating a database based on the real research done, and then identifying, okay, let's see what is binding. So these are experiments done, what is binding, and this experiment, there must be experiments done that show androgen receptor binds to this protein. There are databases. What these databases may be, they may be mass spectrometry databases that suggest binding, and there are text mining as well. So evidence, people making meta-analysis. There are overwhelming evidence to suggest that androgen receptor bind to this protein of interest. So there are few things that you can do at the very early stage in your project to make sure in the end you will have a very robust, meaningful project that may be helpful in saving lives of cancer patients. So with this, I'm going to stop and take any questions if there are any. So I'll give 10-15 minutes now. I didn't give the break before. To those of you who want to stay behind and ask me questions. Swati, are there any questions?
There is one by Padmavati Ganeshan.
Okay, I can't see. Yeah. Okay, yeah, she's asking a question. Sorry, I missed that. Can we use the data from these sites for publishing papers? So I already gave the answer for that. Looks like there aren't any more questions. In which case, shall we close the session, Swati?
Sure, sir. Sure. Thank you very
Okay,
much.
obviously, thank you for your time in
We just
organizing
got
this.
another question.
There is another question. Hamida is asking, Walaikum Salam. I've recently completed Masters in Biochemistry. I have only research experience of dissertation. Do I need to solve more labs or can directly apply for PhD? Do you mean a PhD abroad, outside India, or do you mean in India? Uh, uh, Let me answer Hamida's question. First of all, I'll come back to you. Basically, if you want to do PhD in a good place abroad, as an Indian national, your best bet is if you want to save time and not spend multiple years doing coursework and go to America, which is of course not very permissive these days, as you might have seen. I would say Europe is a very good place to do a PhD, and lots of students go there. If you have, so for example, when I went to do my PhD 22 years ago, they did ask me. The form asked me whether I had qualified GATE and NET because they do. Even back then, they recognize those qualifications as being very competitive. So there were, of course, a preference given to students who have qualified these exams. So that was one element. I had written a project on androgen receptor 1 year before, of course, so I knew about androgen receptor a lot. I did not have very much lab exposure on working on androgen receptor, so I mitigated that not having the research experience by writing by writing the research grant application uh, and submitted it with my future supervisor. That's what I did, and that was successful. But if you're asking me with regards to you completed a master's in biochemistry, have research experience in dissertation, see if it is possible for you to publish a paper from your master's work. I'm not talking about a first author paper, but I'm talking about paper as a collaborative research with other people from your master's lab, perhaps a research article which you may have published. This might be little bit helpful. Certainly no issues with that. But if you want to do a PhD in India, certainly having a JRF would help. I know there are still lots of competition with JRF, that even after JRF, students may not get top labs that they are after. For that, what I would suggest people should do is to join as a research assistant in a top lab. A lot labs that publish very good papers and where they will not be cleaning the glassware, but they will be doing some research for a year or so. And that means if they've done this, they have substantial research experience. They can they can apply extensively. For example, countries like Germany, Belgium, France, to do a PhD there. Um, if you publish any papers or have substantial experience, I get lots of CVs from many countries, including India. And the first thing you see is, okay, you've done CRISPR, you've done Western, I believe you've done Western. I don't think you're an expert of CRISPR. Oh, we have done RNA sequencing. Well, I don't really, because there is no evidence, there is no publication which will substantiate what this student is telling me. I take that with a pinch of salt, and there are plenty of people, sadly, who can write references without full honesty, and that's why you have to be very careful because it is a big commitment taking a student on board. So if you attach yourself, there are institutions in India, the NCBS, IITs, and of course AMU, IIT, IISERs, where there are people doing excellent research. Become part of them. If there is any position for a year, engage with the research, publish some papers, and that will mean by investing that time, even if they're on a voluntary basis, if you invest 1 year, after that you work very super hard, chances are that you will be able to then apply and be expected to get shortlisted at least for the interview. Then it is up to you if you're able to answer questions in an interview, if you talk and if you don't talk. That's why I'm asking you to ask questions to me while you're here, because that helps certainly prepare you for an interview. So I have another question from Mustafa. What is the future of AI in this field? Well, AI has limitations. Remember, it works on machine learning. What you feed AI, it brings you that out. It does not have a brain of its own. So no way this can come anywhere close to human intelligence. It can certainly facilitate research. For example, the platforms I was showing you, many of them are built on machine learning. So certainly that helps, but there is no equivalent to human brain and what it can achieve. Certainly with AI, it might become, you know, relatively faster to do research, but when it comes to doing groundbreaking research, which will change the course of treatment, for example, for cancer, it is going to be largely dependent on uh, genuine thinking, intelligence, and hard work. Uh, Outside, yeah, Hamida, I hope I've answered your question. I'm also working on a dissertation paper. Yes, Hamida, make sure that you publish a paper in a journal which is indexed on PubMed. As a first author, that will be important. That will at least hopefully get you shortlisted. I can give you some idea. I think I applied for 1,700 places when I was a student. Uh, and got shortlisted for few. But one person did offer. There are lots of students applying, especially from East Asia, to come to European Union countries, and that's why there is a huge competition. Sometimes, I mean, even for my lab, you see students who, as a BSc students, have published papers, which is, of course, you can see how high the bar is being set. I'm not saying don't apply to UK, do apply to UK as well, but mostly to positions, for example, in labs which are Wellcome Trust funded or funded by Cancer Research UK, because they provide fee for international students. Sadly, most of the PhD studentships in this country do not provide tuition fee for international student, which is normally 3 to 5 times higher than the fee for British nationals, home students. So, are there any more questions, or shall we close this session? I don't see any more questions, Swati.
Okay, sir. Thank you, sir. Thank you very much for the
Thank
excellent and
you.
insightful lecture and the tutorial.
Thank
So,
you very
with
much.
yes, sir.
Yeah, go on.
Okay. So with this, we conclude today's session. Thank you to all the speakers and participants for your valuable contributions and active participation. And we look forward to seeing you tomorrow.
Thank you, everyone. Take care. Have a
Thank
lovely
you.
evening. Bye.